Beneath every forest, every meadow, every houseplant sitting quietly by a window, something is already happening, not loudly, not quickly, and not in a way that draws attention to itself. Plants appear self-contained. They grow upward, unfold leaves, follow the sun, and seem to manage perfectly well on their own. But that appearance is carefully misleading. Almost everything a plant does above ground is supported by relationships taking place below it, relationships that began hundreds of millions of years ago, relationships that never stopped. Roots do not simply push into soil. They enter a living environment shaped by countless microscopic interactions. Threads thinner than a human hair move through mineral grains and pockets of air, dissolving rock, transporting nutrients and quietly extending the reach of plants far beyond what roots alone could ever achieve. This partnership is ancient, patient, and largely invisible. It does not rely on awareness, intention, or communication in any familiar sense. It works because physics allows it to work, because chemistry makes it possible, and because evolution kept it when nothing better came along. What looks like independence is, in reality, cooperation refined over deep time. And once this hidden partnership becomes visible, it becomes difficult to look at plants the same way again. Welcome to the Sleepy Scientist. If you enjoy calm, thoughtful science like this, take a moment to like the video and subscribe to the channel. And while you settle in, feel free to share where you're watching or listening from, whether that's above a forest floor, inside a city, or somewhere with soil, quietly doing its work beneath you. A plant looks complete when seen from above. A stem rises, leaves spread, light is captured. Growth unfolds in a steady, confident direction. From this angle, a plant appears to be a single, self-contained organism, quietly performing all the tasks required to stay alive. It gathers energy from sunlight, builds its own tissues, and stands in place without needing to move. But this picture is incomplete. What appears to be a whole organism is in reality only the visible portion of something much larger. The plant body does not end at the surface of the soil. It fades into it. And once attention shifts below ground, the idea of a plant as a single, independent individual begins to quietly fall apart. Modern biology increasingly describes many organisms not as isolated entities, but as biological composites. Systems made up of multiple species whose lives are so tightly interwoven that separating them becomes more a matter of definition than reality. One of the most influential frameworks for this idea is the concept of the holobiont. The term holobiont refers to a host organism together with all of its associated microorganisms, functioning as an ecological and physiological unit. This perspective was strongly developed by the evolutionary biologist Lynn Margulis, whose later work on symbiosis extended far beyond the well-known examples of mitochondria and chloroplasts inside cells. Margulis argued that cooperation between distinct life forms was not a rare exception in evolution, but one of its primary creative forces. From this viewpoint, a plant is not simply a plant. It is a composite system made of plant cells, bacterial communities, fungal partners, and a surrounding environment that actively participates in its survival. Nowhere does this become more evident than at the root-soil interface. Roots are often described as organs that absorb water and nutrients from the ground. That description is technically correct, but deeply misleading. Roots alone are remarkably limited in what they can do. They grow slowly through dense material. Their surface area is restricted. Many essential nutrients, especially phosphorus and certain forms of nitrogen, move very poorly through soil. Without assistance, roots struggle to access them in meaningful quantities. This is where individuality begins to break down. The narrow region where roots meet soil is one of the most biologically active environments on land. It is known as the rhizosphere, and it is crowded with bacteria, fungi, archaea, and microscopic animals. Chemical signals flow constantly. Sugars leak from root cells. Enzymes reshape minerals. Entire metabolic pathways unfold across species boundaries. In this zone, it becomes difficult to say where the plant ends and the environment begins. Plants actively release a portion of the sugars they produce through photosynthesis into the soil. This process is known as root exudation. Measurements suggest that between 10 and 40% of a plant's photosynthetically fixed carbon can be transferred below ground in this way. From a purely individualistic perspective, this looks inefficient, even wasteful. But the plant is not giving this energy away into emptiness. Those sugars feed dense microbial communities that, in turn, alter the chemistry and structure of the soil. Bacteria fix nitrogen, converting it from inert atmospheric forms into compounds plants can use. Fungi extend thread-like filaments far beyond the physical reach of roots, scavenging nutrients from microscopic cracks in rock and soil particles. Together, these organisms create a living interface that dramatically expands what the plant can access. At this point, the idea of a plant as a bounded individual becomes hard to defend. Fungal partners, especially mycorrhizal fungi, do not merely assist plants from the outside. They integrate directly into root tissues, forming structures that blur the distinction between plant and fungus. In arbuscular mycorrhizal associations, fungal filaments penetrate root cells and form branching structures that greatly increase the surface area for nutrient exchange. These structures are temporary, continuously built and dismantled as the partnership persists. The plant cell remains intact, the fungus remains genetically distinct, but functionally they operate as a shared system. Plant ecology research has repeatedly shown that the vast majority of land plants form mycorrhizal associations. Estimates suggest that over 90% of plant species rely on fungal partners to some degree. Plants that do not form these relationships are unusual, often occupying very specific ecological niches or relying on alternative strategies that come with significant trade-offs. From this perspective, mycorrhizal partnership is not an optional enhancement. It is the default condition for plant life on land. This challenges a deeply ingrained intuition about biology. There is a strong tendency to imagine organisms as discrete units that interact with their surroundings but remain fundamentally separate from them. This intuition works reasonably well for large animals with clear physical boundaries. It works less well for plants, and it breaks down almost entirely once microbial and fungal partners are included. A plant's physiology extends outward through fungal networks and microbial processes. Its metabolism does not stop at its own cells. Nutrient acquisition, stress tolerance, and even disease resistance depend on organisms that are not genetically part of the plant itself. Modern plant microbiome research has reinforced this view. Studies using genetic sequencing have revealed complex structured microbial communities associated with plant roots, leaves, and internal tissues. These communities are not random. Different plant species consistently associate with particular microbial groups. Changes in these communities can alter plant growth, flowering time, and survival under stress. In other words, part of what makes a plant the kind of plant it is exists outside its own genome. This does not mean that plants are helpless or incomplete in a negative sense. It means they are distributed. Their functional bodies are spread across multiple organisms and physical spaces. The plant that appears above ground is a visible node in a much larger biological network. Seen this way, The idea of plant independence becomes a convenient simplification rather than an accurate description. Even the most basic acts of plant life rely on this extended system. When a plant absorbs phosphorus, it is often receiving a molecule that has been liberated from mineral surfaces by fungal enzymes. When it acquires nitrogen, that nitrogen may have passed through bacterial metabolism before ever entering a root. When it tolerates drought, the structure of the surrounding soil, shaped by microbial activity, plays a decisive role in water retention. None of this requires awareness, intention, or coordination in any cognitive sense. It arises from physical proximity, chemical compatibility, and evolutionary persistence. Over hundreds of millions of years, plants that engaged effectively with surrounding life survived and spread. Those that did not were quietly filtered out. The result is a form of life that cannot be cleanly separated from its context. There is a gentle irony here. Plants are often described as simple organisms, rooted, passive, lacking complexity compared to animals. Yet their mode of existence depends on managing relationships across multiple kingdoms of life simultaneously. Their simplicity above ground is supported by extraordinary complexity below it. The plant body, then, is not confined to stems, leaves, and roots. It includes fungal filaments threading through soil, bacterial colonies cycling nutrients, and chemical gradients shaped by countless interactions. The visible plant is only the most obvious expression of this system. Once this perspective settles in, it becomes difficult to look at a plant and see a single organism standing alone. What stands instead is a collaboration refined over deep time, quietly functioning without central control, without boundaries that can be neatly drawn, and without any need to announce itself. The plant, as it turns out, is larger than it looks. Fungi are often introduced to us through their endings: rotting logs, softening fruit, the slow return of fallen leaves to soil. In this familiar role, fungi appear as nature's clean-up crew, quietly breaking down what is no longer alive. Useful, certainly, essential, even, but fundamentally secondary. Life finishes, and fungi arrive to tidy the remains. This picture is accurate, but it's far from complete. In reality, fungi are not simply agents of decay. They are among the most important interfaces between the non-living world of minerals and the living world of metabolism. They operate at a boundary that plants alone cannot cross efficiently, translating raw chemistry into biological possibility. To understand this role, it helps to step back and consider the basic problem of life on land. Most of the elements required for living tissue exist abundantly in Earth's crust. Phosphorus, potassium, calcium, iron, and many trace metals are everywhere. The difficulty is not their presence, it is their accessibility. Locked inside mineral lattices or bound tightly to soil particles, these nutrients do not drift freely toward roots. They diffuse slowly, if at all. From the perspective of a plant root, the surrounding soil is rich but frustratingly unreachable. Fungi evolved to live precisely in this gap. Unlike plants, fungi do not photosynthesize. They do not capture energy directly from sunlight. Instead, they grow as vast networks of microscopic filaments called hyphae, each one only a few micrometers thick. These filaments branch repeatedly, forming mycelial networks that can extend for meters, kilometers, or more, depending on conditions. This growth form is not accidental. It is an elegant response to physical constraints. Diffusion, the passive movement of molecules from areas of high concentration to low concentration, is efficient only across very short distances. Beyond that, it becomes painfully slow. For a root, increasing access to distant nutrients by simple diffusion would require enormous surface area and energy investment. Fungi solve this problem by becoming surface area. By spreading thin filaments through soil pores and microscopic cracks, fungal networks massively increase the contact area between living tissue and mineral surfaces. The economics of surface area matter here. A structure that is thinner can explore more space using the same amount of material. This principle, known in biology as surface area to volume optimization, underlies everything from lung alveoli to leaf veins. Fungi apply it underground with extraordinary efficiency. Once in contact with minerals, fungi deploy chemistry. They secrete organic acids and enzymes that alter the local environment, dissolving mineral bonds and freeing nutrients that would otherwise remain locked away. Phosphorus is a particularly important example. Although abundant in soils, it is often trapped in insoluble forms. Fungal enzymes convert it into forms that can enter biological pathways. At this point, fungi have done something remarkable. They have taken matter that was geologically present but biologically inaccessible, and moved it across the boundary into metabolism. This is why it's more accurate to think of fungi as environmental interfaces rather than assistants or helpers. They occupy a position between worlds. On one side lies rock, mineral and chemistry governed by slow geological processes. On the other lies living tissue, fast turnover and biological demand. Fungi bridge these domains continuously. This framing has been strongly emphasized by contemporary fungal biologists, including Merlin Sheldrake, whose work focuses on fungal physiology and ecological function rather than metaphorical interpretations. In his research and synthesis of existing literature, fungi emerge not as background organisms but as central mediators of terrestrial life. Their power lies not in communication or intelligence, but in physical reach and chemical versatility. Importantly, this mediation is not performed with plants specifically in mind. Fungi did not evolve to serve plants, they evolved to survive. Their ability to access minerals, exploit small spaces, and metabolize complex substrates benefits them directly. When plants entered this system, a relationship formed because the needs of each overlapped. Plants possessed carbon-rich compounds produced through photosynthesis. Fungi possessed access to soil-bound nutrients. Exchange followed naturally. Calling this a partnership is accurate, but it risks understating the deeper role fungi play. They are not optional collaborators that plants occasionally rely on. They are structural components of the environment itself. From the perspective of a plant root, fungi effectively redesign the soil. They alter nutrient gradients, water movement, and chemical availability. They change what the environment offers before the plant ever responds. Water provides another clear example. Fungal hyphae can access thin films of water held tightly between soil particles, water that roots alone cannot easily reach. By moving this moisture along their networks, fungi buffer plants against short-term drought. The effect is not dramatic in a single moment. It is cumulative, subtle, and enormously important over time. Measurements in forest soils show that mycorrhizal networks can extend the effective absorptive area of a root system by several orders of magnitude. A single plant may be connected to meters or kilometers of fungal filament. Each filament is alive, each is metabolically active. Together they form an extension of the plant's interface with its surroundings. Again, this is not coordination in a cognitive sense. It is physics and chemistry operating through living structures. There is a temptation to describe fungal networks as intelligent or purposeful. This temptation arises because their outcomes appear organized. Nutrients move efficiently. Resources are redistributed. Systems remain stable across decades. But no planning is required. What emerges instead is constraint-driven order. Diffusion limits surface area economics, and chemical affinities shape what is possible. Evolution retains configurations that persist. Over time, what remains looks deliberate, even though it arises without intention. Understanding fungi in this way dissolves another common misconception. They are not merely partners that plants recruit when conditions are poor. They are constant mediators that define baseline conditions under which plants exist at all, even when plants appear to grow without fungal assistance. Fungal influence often remains present in the background, shaping soil structure and nutrient availability long before roots arrive. Seen from this angle, fungi become something closer to infrastructure than organism. They are not passive scenery. They are active systems that maintain the conditions life depends on. The quiet humour here is that fungi are among the least conspicuous forms of life. They rarely draw attention above ground. Their most extensive bodies are hidden entirely from view, and yet, without them, terrestrial ecosystems would rapidly lose coherence. Soil would compact, nutrient cycles would stall, plant growth would become brittle and inefficient. Life would not vanish overnight, but it would slow, thin, and simplify. Fungi prevent this outcome not by dramatic intervention, but by continuously translating between worlds. They take the slow language of geology and convert it into the fast language of biology. They allow living systems to tap into the deep reserves of the earth without waiting for rock to weather on its own timescale. Once this role is recognized, it becomes clear that fungi are not supporting actors in the story of plants. They are the medium through which plants interact with the planet itself. They are not partners of convenience. They are the environment, made alive. For a long time, evolution has been told as a story of rivalry. Organisms compete for space, for light, for nutrients, for survival itself. Those that outcompete their neighbours persist, while others fade away. This framing is not wrong. Competition is real, measurable, and powerful. But when it becomes the dominant lens through which all of evolution is interpreted, something important is lost. Life did not only advance by pushing others aside, it also advanced by joining forces. The word used to describe this joining is symbiosis. It was formally introduced in the late 19th century by the German botanist Anton de Bari. De Bari defined symbiosis simply as the living together of unlike organisms. The definition was deliberately neutral. It did not imply cooperation, harmony, or benefit. It described proximity and persistence, two different life forms remaining together over time. That modest definition turned out to be quietly radical. At the time, biology was still deeply influenced by ideas of independence and hierarchy. Organisms were expected to function as self-contained units, interacting with others only at their boundaries. De Bari's framing suggested something else entirely. It allowed for the possibility that long-term association itself could shape what an organism becomes. Over the following decades, symbiosis was often treated as an oddity, a curiosity. Lichens, for example, were recognized as stable associations between fungi and photosynthetic partners, but they were viewed as special cases rather than clues to a broader pattern. The main current of evolutionary theory continued to emphasize competition, gradual change, and individual selection. It was not until much later that this balance began to shift. Historians of biology such as Jan Sapp played a crucial role in revisiting how symbiosis had been understood, and in many cases marginalized. Sapp's work traced how cooperative and symbiotic ideas repeatedly surfaced in biology, only to be pushed aside when they conflicted with more dominant narratives. His analyses showed that symbiosis was not ignored because it lacked evidence, but because it did not fit comfortably within prevailing frameworks. When symbiosis is taken seriously, evolution looks different. Instead of seeing organisms as isolated competitors occasionally interacting, evolution begins to resemble a process of integration. New forms arise not only through modification of existing structures, but through the merging of capabilities across species boundaries. Stability becomes as important as advantage. Persistence matters as much as speed. This shift in perspective has been reinforced by some of the most fundamental discoveries in modern biology. The clearest example is endosymbiosis, the process by which entire organisms became incorporated into others. Mitochondria, the structures inside cells that generate usable energy, originated as free-living bacteria. Chloroplasts, which allow plants to perform photosynthesis, also began as independent organisms. These events did not happen repeatedly or casually. They occurred rarely, under specific conditions, and once established, they reshaped the trajectory of life on Earth. These are not marginal details. Without them, complex life as we know it would not exist. What is often overlooked is that symbiosis did not stop there. Plant-fungus relationships represent another layer of evolutionary integration, operating not inside cells but across tissues, organisms, and environments. These relationships are not temporary alliances. They are ancient, stable, and remarkably resistant to disruption. Fossil evidence suggests that mycorrhizal associations were present when plants first colonized land more than 400 million years ago. Early plant fossils show structures consistent with fungal partnerships, indicating that plants did not conquer land alone. They arrived already connected. This persistence matters. Evolution tends to discard traits that are costly without delivering consistent benefit. Structures that persist across hundreds of millions of years through mass extinctions, climate shifts, and continental rearrangements are not evolutionary accidents. They are foundational solutions to enduring problems. Plant-fungus symbiosis solves several of those problems at once. It addresses nutrient limitation in soils where diffusion is slow and minerals are inaccessible. It buffers water availability in environments prone to drought. It stabilizes growth across variable conditions. And it does so without requiring plants to evolve entirely new organs or metabolic pathways on their own. From an evolutionary standpoint, this is efficient. Rather than reinventing chemistry, plants integrated with organisms that had already mastered it. Rather than expanding roots endlessly, they partnered with networks that could explore soil more effectively. Selection favored plants that participated in these relationships, not because cooperation was virtuous, but because it worked. This challenges the idea that symbiosis is a secondary feature layered onto otherwise independent organisms. In the case of plants and fungi, symbiosis is not an add-on. It is a core condition of existence. The plant lineage did not evolve first and later discover fungal partners. The two histories are entangled from the beginning of terrestrial life. Competition still operates within this system. Plants compete for light. Fungi compete for resources. Different fungal species compete for access to roots. But this competition unfolds inside a cooperative framework that remains intact. This layered structure is important. Evolution is not a simple contest where cooperation replaces competition or vice versa. It is a nested system. Cooperation stabilizes relationships that persist long enough for competition to refine them. Symbiosis creates the stage on which selection can act. Jan Sapp and others highlighted that ignoring this structure leads to an incomplete understanding of evolution. When focus remains fixed on individuals and rivals, long-term integration disappears from view. Yet integration is precisely what allowed life to increase in complexity without collapsing under its own demands. The persistence of mycorrhizal associations illustrates this beautifully. Across modern ecosystems, from tropical forests to grasslands to boreal soils, mycorrhizal fungi remain deeply embedded in plant life. Different forms exist. Arbuscular mycorrhizae dominate in many herbaceous plants. Ectomycorrhizae are common in forest trees. The details vary, but the principle remains constant. Plants rarely walk away from this relationship. Even in nutrient-rich environments where fungal assistance might seem unnecessary, plants often retain mycorrhizal partners. The benefits may shift. Stress tolerance, disease resistance, or long-term soil stability may become more important than immediate nutrient gain. But the relationship endures. This endurance is a strong signal that symbiosis is not a temporary evolutionary shortcut. It is a stable solution. Seeing symbiosis as central rather than exceptional also helps explain why purely competitive models struggle to account for real ecosystems. Forests do not behave like collections of isolated individuals locked in constant conflict. They behave like systems with shared constraints, shared resources, and shared histories. Symbiosis provides the connective tissue that makes this possible. Once this is acknowledged, evolution begins to look less like a battlefield and more like a long conversation between life forms, mediated by physics, chemistry, and time. Some voices fade, others merge. A few persist for hundreds of millions of years, not because they dominate, but because they fit together. Plant-fungus symbiosis is one of those enduring conversations. It is not an evolutionary footnote. It is a recurring theme, written into the structure of terrestrial life. And it reminds us that some of the most powerful forces shaping evolution are not conflicts that end in winners and losers, but relationships that simply refuse to fall apart. When mycorrhizae are first introduced, they are often framed as clever additions, helpful extras, optional upgrades, something plants can make use of when conditions are poor. This framing is comfortable, but it quietly reverses the truth. Mycorrhizae are not special adaptations that some plants happen to possess. They are the baseline condition for plant life on land. To understand plants without them is to start from an exception and mistake it for the rule. Ecological surveys across continents and ecosystems have repeatedly shown that the majority of plant species form mycorrhizal associations. Conservative estimates place the figure above 90%. In forests, grasslands, wetlands, and even many agricultural soils, mycorrhizal fungi are present wherever plants grow naturally. Different environments favour different fungal groups, but the underlying relationship remains remarkably consistent. This is not a marginal pattern, it is a dominant one. From this perspective, a plant without mycorrhizal partners is unusual. Such plants exist, but they are ecological specialists. They often occupy disturbed habitats, nutrient-rich soils, or environments shaped by intense selection pressures that favour rapid growth over long-term stability. Even then, their independence often comes with costs. The more common condition is interdependence. Framing mycorrhizae as a default state rather than an adaptation changes how plant biology is understood. Instead of asking why some plants rely on fungi, a more revealing question emerges. Why would any plant attempt to grow without them? To answer that, it helps to consider what mycorrhizae actually provide. As living networks embedded in soil, mycorrhizal fungi extend the effective reach of plant roots by orders of magnitude. A root system that might physically occupy a few cubic centimeters of soil can, through fungal filaments, access volumes hundreds or thousands of times larger. This expansion does not require thicker roots or greater energy expenditure from the plant itself. It relies on fungal growth strategies already optimized for navigating complex, compact environments. In return, plants provide fungi with a steady supply of carbon compounds derived from photosynthesis. This exchange is not occasional, it is continuous. Carbon flows downward into fungal networks as long as photosynthesis continues above ground. Nutrients flow upward as long as fungal metabolism remains active. Over time, this mutual exchange becomes embedded in plant development itself. Roots of mycorrhizal plants often grow differently from those of non-mycorrhizal species. They may produce fewer fine root hairs, relying instead on fungal filaments to perform that function. Root architecture, branching patterns, and even growth rates can be shaped by the presence of fungal partners. This is not dependence in the sense of weakness, it is condition dependence. Condition dependence describes a state in which an organism evolves within a context that reliably provides certain functions externally. Over generations, traits that duplicate those functions internally may be reduced or lost, not because the organism is deficient, but because maintaining redundant systems is costly. Efficiency, not fragility, drives the process. A familiar analogy exists inside cells. Mitochondria perform energy conversion so effectively that cells no longer maintain independent pathways for generating the same energy yield. The cell becomes dependent, but also vastly more capable. The same logic applies at the plant-fungus interface. Plants that evolved alongside mycorrhizal fungi did not need to solve the problem of mineral extraction alone. They evolved roots that cooperate rather than roots that conquer. This perspective reframes what it means for a plant to be normal. Normal plant life is not solitary, it is relational. A plant growing in isolation, without microbial or fungal partners, is experiencing a simplified version of its ecological identity. Such a plant may survive, especially under artificial conditions, but it's not expressing the full range of capabilities shaped by evolution. This becomes clear in controlled experiments. Plants grown in sterile soils, without microbial communities, often display stunted growth, altered nutrient profiles, and increased sensitivity to stress. Reintroducing mycorrhizal fungi frequently restores growth patterns closer to those seen in natural environments. These results have been documented repeatedly in plant ecology and soil science research, not as anomalies, but as expected outcomes. The absence of mycorrhizae is not neutral. It is a significant deviation from ecological norm. Non-mycorrhizal plants illustrate this deviation clearly. Many belong to plant families that evolved specific strategies to bypass fungal dependence. Some produce dense root hairs and high concentrations of enzymes to mobilize nutrients directly. Others thrive in environments where nutrients are freely available, such as disturbed soils rich in nitrogen. These strategies work, but they are narrow. They tie plants to particular conditions and reduce flexibility. Mycorrhizal plants, by contrast, remain adaptable across a wider range of environments because their fungal partners mediate local conditions on their behalf. This difference helps explain why mycorrhizal associations persist across geological time. Fossil evidence indicates that early land plants formed mycorrhizal relationships from the outset. Over hundreds of millions of years, through shifts in climate, atmospheric composition, and soil structure, this association remained intact. It adjusted, diversified, and specialized, but it did not disappear. Evolution rarely preserves fragile arrangements for that long. The language of dependence often carries negative weight, suggesting vulnerability or limitation. In biology, however, dependence frequently signals stability. Systems that persist do so because their components reliably meet one another's needs. Condition dependence reflects trust shaped not by intention, but by consistent outcomes across generations. Plants trust mycorrhizal fungi not because fungi are benevolent, but because the partnership works often enough, across enough contexts, to remain favoured by selection. Once this is recognised, the idea of a plant standing alone begins to feel like a conceptual convenience rather than a biological reality. What appears to be a self-sufficient organism is better understood as a hub, connected to networks that extend beyond its tissues. This shift in perspective is subtle, but it matters. It encourages a view of life that prioritises relationships over boundaries, processes over objects, and systems over individuals. It invites a quieter appreciation of how life persists not through independence, but through reliable connection. In this light, mycorrhizae are not remarkable add-ons to plant life. They are the context in which plant life makes sense at all. What looks like dependence is simply how plants have always lived. Metabolism is often described as something that happens inside an organism. Sugars are made in leaves, energy is used in cells, waste is managed internally. From this viewpoint, each living thing appears to run its own biochemical economy, producing what it needs and spending what it has. That picture is tidy. It is also incomplete. In many ecosystems, metabolism is not confined to single bodies. It is distributed across species, shared among partners whose biochemical roles interlock so closely that no single organism completes the full process alone. What looks like individual metabolism is in practice a collective one. Plants and fungi provide one of the clearest examples of this arrangement. Consider carbon, the backbone of biological life. In plants, carbon enters the system through photosynthesis. Light energy is used to convert carbon dioxide into sugars, which then serve as both building material and energy source. This step is uniquely plant-driven. Fungi cannot do it. Bacteria cannot do it in the same way. Without plants, this carbon remains inaccessible to most terrestrial life. But once carbon has been fixed into sugar, the story immediately stops being private. A significant fraction of that newly made carbon is transported downward, away from leaves and stems and into roots. From there, it moves outward into the surrounding soil through a process known as root exudation. Sugars, amino acids, organic acids, and a wide range of secondary metabolites leak, or are actively released from root tissues into the rhizosphere. Measurements using carbon isotope tracing have shown that between 10 and 40% of a plant's photosynthetic output can enter the soil in this form. The exact amount depends on species, growth stage, nutrient availability, and environmental stress. Even at the low end, this represents a substantial investment. This carbon does not vanish. It is taken up almost immediately by fungi and bacteria living in close association with the roots. For these organisms, plant carbon is not a bonus. It is their primary fuel. Fungal metabolism in particular depends on this steady flow. In exchange, fungi mobilize nutrients, transport water, and chemically modify the soil in ways that feed back into plant growth. From a biochemical perspective, this is metabolic pathway partitioning. No single organism performs the entire sequence from raw materials to functional outcome. Plants specialize in carbon fixation. Fungi specialize in mineral extraction and transport. Bacteria contribute nitrogen transformation, hormone production, and enzymatic diversity. Each partner performs a subset of reactions, and the overall metabolic task is completed only when these subsets are connected. This idea is central to biochemical ecology. a field that studies how metabolic processes operate across ecological relationships rather than within isolated organisms. Instead of asking what a plant does on its own, biochemical ecology asks what becomes possible when organisms metabolize together. Seen through this lens, the plant-fungus relationship is not an act of generosity. It is an integrated metabolic system. Carbon flow provides a clear illustration. Once carbon enters fungal networks, it does not simply remain there. Fungi respire some of it, releasing carbon dioxide back into the soil atmosphere. Some is incorporated into fungal biomass. Some becomes part of complex soil organic matter, bound to mineral surfaces and protected from rapid decomposition. This redistribution has measurable consequences. Soils associated with active mycorrhizal networks often retain carbon for longer periods, contributing to long-term carbon storage. Studies in forest ecosystems suggest that mycorrhizal activity influences how much carbon remains underground versus returning quickly to the atmosphere. The effect is not dramatic on short timescales, but over decades and centuries, it shapes ecosystem carbon balance. Importantly, plants do not direct this process in any deliberate way. Carbon moves along gradients of concentration and demand. Fungal metabolism draws carbon toward itself because it needs energy. Plant roots release carbon because internal concentrations and transport dynamics make release inevitable. What emerges is a stable exchange shaped by physical constraints and evolutionary history, not intention. This stability has been studied extensively by soil scientists such as Yakov Kuzyakov, whose work on rhizosphere processes and carbon flux has helped quantify how plant-derived carbon moves through soil systems. Using isotope labeling techniques, researchers have traced carbon atoms from leaves into fungal tissue, microbial biomass, and soil organic matter within days. These findings reinforce a key point. Plant metabolism does not end at the root surface. It diffuses outward, becoming inseparable from the metabolism of surrounding organisms. Root exudation dynamics illustrate this further. For a long time, exudation was thought to be a passive leak. Sugars escaping damaged cells, excess carbon spilling over. More recent work has shown that plants actively regulate exudation rates in response to nutrient availability and microbial presence. Different compounds are released under different conditions, shaping which microbial communities flourish nearby. Plant physiologists such as Hans Lambers and Ian C. Anderson have studied how nutrient limitations influence carbon allocation below ground. Their work demonstrates that plants increase exudation when phosphorus or nitrogen becomes scarce, effectively investing carbon to recruit microbial assistance. Again, this is not altruism. From a systems perspective, sharing carbon improves overall efficiency. The plant gives up some immediate energy in exchange for access to resources it cannot otherwise obtain. The fungal network expands its energy supply, and in doing so maintains the very soil conditions that support plant growth. Metabolic sharing reduces redundancy. If plants attempted to perform all these functions internally, they would need far more complex root systems, additional enzymes, and higher energy expenditure. By offloading part of the metabolic workload to fungi and microbes, plants remain structurally simpler and energetically leaner. This division of labour enhances stability. When environmental conditions fluctuate, different parts of the system respond at different rates. Microbial communities can adjust rapidly. Fungal networks can shift resource flows. Plants can alter carbon allocation. The result is a buffered response to stress, smoothing out extremes that would otherwise disrupt growth. This buffering effect is one reason mycorrhizal plants often show greater resilience to drought, nutrient depletion, and soil disturbance. The shared metabolic system absorbs shocks more effectively than any single organism could. There is a subtle humour in how this works. Plants are often imagined as stoic individuals, silently enduring whatever the environment delivers. In reality, they constantly outsource critical metabolic tasks to organisms that are far more chemically versatile. What looks like quiet endurance is supported by busy underground trade. The language of sharing can sound moral, but in biology it is mechanical. Carbon flows where it is used. Nutrients move where gradients allow. Stability emerges because the system has a fewer bottlenecks and more pathways. This is why distributed metabolism persists. It is not maintained because organisms are cooperative in a social sense. It is maintained because systems that distribute metabolic work are more robust. They fail less often. They recover more quickly. They persist across changing conditions. When metabolism is shared, life becomes harder to disrupt. Seen this way, the plant-fungus relationship is not a story about generosity or sacrifice. It is a story about efficiency achieved through connection. By spreading metabolism across species, life avoids the fragility that comes with doing everything alone. The plant makes sugar, the fungus moves minerals, the soil stores memory. Together they form a metabolic system larger than any one of them. And once metabolism is understood as something that flows across boundaries, rather than staying neatly inside them, the idea of an organism as a closed biochemical unit begins to quietly dissolve. When people first encounter the underground world of plants and fungi, the language that comes most easily is the language of communication. Signals are sent, messages are received, information travels, decisions are made. This way of speaking is understandable. Patterns appear coordinated. Resources move where they are needed. Systems respond to change in ways that feel purposeful. From the surface, it can look as though something must be directing the flow, but this impression comes from a familiar habit of thought rather than from how the system actually works. Coordination in living systems does not require messaging, intention, or control in any human sense. Order can emerge without instructions. Information can exist without signals. Direction can arise without a director. To see how, it helps to step outside biology for a moment and look at the physics that underlies all living processes. Living systems exist far from equilibrium. This means they are constantly exchanging energy and matter with their surroundings. They are not static objects settling into rest. They are ongoing processes maintained by flow. This idea was formalized in the work of the physicist and chemist Ilya Prigogine, who studied what he called dissipative structures. These are ordered patterns that arise spontaneously in systems driven by energy flow. Examples include convection cells in heated fluids, chemical oscillations, and atmospheric circulation patterns. None of these systems are controlled from above. No molecule knows the shape the pattern will take. The order appears because energy is moving through the system, and constraints shape how that movement can occur. Life operates in exactly this regime. Plants, fungi, and soil organisms form open systems through which energy and matter constantly pass. Sunlight enters as energy. Carbon dioxide enters as raw material. Water and minerals flow in from the environment. Heat and waste flow out. As long as these flows continue, structure persists. Within this context, coordination does not need to be sent, it emerges. Consider how fungal networks grow. Hyphae extend outward following gradients of nutrients and moisture. Growth is faster where resources are abundant and slower where they are scarce. Branching occurs when it improves access. Retraction occurs when it does not. No signal is sent announcing where to grow next. The physical environment itself provides the information. Information, in this sense, is not a message, it is a constraint. A gradient of phosphorus in soil constrains where fungal growth is useful. A local shortage of carbon constrains how far that growth can continue. The resulting pattern reflects the structure of the environment rather than any internal plan. Plants operate in the same way. Roots grow where water and nutrients are more available. Transport within the plant follows pressure gradients and concentration differences. Carbon moves from areas of high production to areas of high demand. This movement is often described as allocation, but it does not require decision-making. It follows the rules of fluid dynamics and chemical potential. When plants and fungi interact, their flows intersect. Carbon released by roots becomes available to fungi. Nutrients mobilized by fungi become available to roots. Water transported along fungal filaments alters moisture gradients in the soil. Each flow reshapes the conditions that guide the next flow. What results is coordination without communication. This is where communication metaphors begin to mislead. It is tempting to imagine fungi sending messages to plants, or plants requesting assistance. But such framing implies symbols, encoding, and interpretation. None of these are required. Instead, coordination arises because both organisms are embedded in the same physical system. They respond to the same constraints, they shape the same gradients, their activities feed back into one another automatically. This does not make the system less remarkable. If anything, it makes it more so. The absence of control does not lead to chaos, it leads to patterned stability. Flows settle into channels that persist because they work. When conditions change, those channels shift. The system adapts not by issuing new instructions, but by redistributing movement. This is what Prigogine meant when he described order emerging from flow rather than being imposed from above. In the soil, energy is constantly dissipated, chemical bonds are broken and reformed, heat is released, matter is rearranged. From this restless background, relatively stable structures arise. Fungal networks maintain their form. Root systems persist. Nutrient cycles repeat. The stability is dynamic. It depends on continued movement. If flow stops, structure collapses. This understanding dissolves another common misconception. The idea that information must travel in order for coordination to occur assumes that information is something separate from matter. In many physical systems, information is embodied in arrangement itself. A river does not signal its banks, yet its shape reflects the constraints imposed by the terrain. Sand dunes do not communicate, yet their ripples align and migrate in patterned ways. In each case, form carries information about forces acting on the system. Living systems work the same way. In plant-fungus systems, information about nutrient availability, water stress, and energy supply is encoded directly in concentrations, pressures, and chemical states. Organisms respond to these states because they cannot do otherwise. Their biochemistry ensures it. This perspective gently dismantles the idea that communication is required for cooperation. Cooperation emerges when multiple processes align under shared constraints. It does not require awareness. It does not require signaling. It requires compatibility. Compatibility, in turn, is shaped by evolution. Over deep time, interactions that destabilize the system were eliminated. Flows that supported persistence were retained. The result is a network of processes that appear coordinated because they have been filtered to fit together. There is a quiet elegance in this. The system does not need to know what it is doing. It only needs to continue doing what works. Patterns that dissipate energy efficiently while maintaining structure endure. Others fade away. This also explains why attempts to assign intention to these systems feel unsatisfying on closer inspection. Intention implies choice among alternatives, but at the level of roots and hyphae, alternatives are constrained long before any choice could be imagined. Growth follows gradients, transport follows pressure, chemistry follows affinity. Wonder does not disappear when intention is removed, it simply shifts. The wonder lies in the fact that complex responsive systems can arise without messages being sent or plans being made. That order can be a byproduct of flow. That stability can be achieved without control. Plants and fungi do not need to talk to coordinate. They are already speaking the same physical language. This understanding also helps clarify why these systems are so resilient. Without central control, there is no single point of failure. Disturb one part, and flows reroute. Interrupt one pathway, and others compensate. The system adjusts because it is governed by constraints rather than commands. This is not intelligence in any familiar sense. It is something quieter, and in many ways more powerful. It is the intelligence of physics given time. Seen this way, the underground world becomes less like a conversation and more like a dance shaped by gravity, chemistry, and energy. No one leads, no one follows, yet the movement remains coherent. Information is present everywhere, not as signals passed along a line, but as the shape of the system itself. And once that idea settles in, the silence beneath our feet begins to feel less empty and more full of structure, quietly holding together, simply by flowing. Soil is often treated as background, something plants sit in, something roots pass through, something that can be scooped, tilled, compacted, or ignored without much thought. This way of seeing soil makes it feel passive, almost inert. A mixture of sand, silt, clay, and organic matter. Useful, certainly, but fundamentally secondary. Life happens above it. Soil simply holds things in place. This impression is deeply misleading. Soil is not a static substrate. It is a self-organizing biological system, shaped continuously by living processes, physical forces, and deep time. It changes slowly, remembers what has happened within it, and responds to disturbance in ways that reveal a long internal history. To understand this, it helps to begin with one of the most influential frameworks in soil science. In the mid-20th century, the soil scientist Hans Jenny formalized what became known as the factors of soil formation. Jenny identified 5 primary influences that determine how soil develops over time: climate, organisms, relief, meaning topography, parent material, the underlying rock or sediment, and time itself. The elegance of this framework lies in what it implies. Soil is not just broken rock mixed with decaying matter. It is the outcome of ongoing interaction between living systems and physical constraints. Of the 5 factors Jenny identified, organisms play a central role. Plants, microbes, and fungi are not merely inhabitants of soil. They are architects. Fungi in particular contribute to soil organization in ways that are easy to overlook because they operate at scales both microscopic and vast. As fungal hyphae grow through soil, they bind particles together into aggregates. These aggregates create pores, channels, and stable structures that influence how air and water move underground. The resulting architecture determines whether soil drains quickly or retains moisture, whether roots can penetrate easily or struggle, and whether nutrients remain available or wash away. This structure is not random. It reflects the cumulative activity of organisms over long periods. Each generation of fungal growth reinforces certain pathways and stabilizes certain arrangements. When hyphae die, their remains become part of the organic matrix, contributing to soil carbon pools and physical cohesion. One of the most studied compounds involved in this process is glomalin, a sticky glycoprotein produced by arbuscular mycorrhizal fungi. Glomalin helps glue soil particles together, increasing aggregate stability. Measurements have shown that glomalin can persist in soils for years or even decades, long after the fungi that produced it have moved on or died. This persistence gives soil a form of memory. Not memory in a cognitive sense, but structural memory. The soil retains traces of past biological activity. Areas that supported dense fungal networks in the past often maintain better structure, higher carbon content, and greater resilience to disturbance long afterward. In this way, soil becomes a historical record of life. Layers of soil, known as horizons, reflect different stages of development shaped by climate, vegetation, and biological activity. A forest soil carries a different signature from a grassland soil. A soil that developed under long-term fungal networks looks and behaves differently from one that has been repeatedly disturbed. These differences are not superficial, they affect how entire ecosystems function. Soil structure influences root growth patterns, water infiltration rates, and nutrient cycling efficiency. Stable aggregates prevent erosion and slow the loss of organic matter. Porous networks allow oxygen to reach roots and microbes, supporting aerobic metabolism. Together, these features create conditions that support sustained plant growth. Remove or disrupt them, and the consequences ripple upward. When soil structure collapses, water either drains too quickly or stagnates, roots suffocate or dry out, nutrients become less available, above-ground vegetation becomes more vulnerable to stress, disease, and collapse. This link between underground organization and above-ground stability is one of the most important and least visible relationships in terrestrial ecosystems. Fungal networks are key to maintaining this link. By continuously exploring soil and binding it together, fungi help maintain a dynamic balance between solidity and permeability. Soil must be firm enough to hold structure, but loose enough to allow movement. This balance emerges through countless small interactions repeated over time. Again, there is no central planner. Soil does not organize itself because organisms intend it to. It organizes because flows of energy, growth, and decay create patterns that persist. Fungi follow moisture and nutrients. Roots follow fungi. Microbes follow carbon. Each response reshapes the environment slightly, altering the conditions for the next response. This is self-organization in its simplest form. Patterns arise because they are reinforced. Structures remain because they channel flow effectively. Inefficient arrangements disappear as conditions shift away from them. Over years, decades, and centuries, these processes produce soils that are finely tuned to local conditions. A hillside soil differs from a valley soil not because someone designed it that way, but because gravity, water movement, and biological activity interacted differently in each place. Hans Jenny's framework captures this beautifully. Climate influences the rate of chemical reactions. Organisms drive biological transformation. Relief shapes water flow. Parent material sets initial constraints. Time allows small effects to accumulate. Fungi sit at the intersection of all these factors. They respond to climate by adjusting growth rates. They interact with plants and microbes. They follow topography through moisture gradients. They weather parent material chemically. And they operate patiently across time. This patience is easy to miss. From a human perspective, soil appears slow, changes are subtle, seasons pass without obvious transformation. But measured across decades, soil systems are remarkably dynamic. carbon accumulates, structure improves or degrades, entire profiles shift. Long-term ecological experiments have shown that soils can take decades to recover from disturbance, even after vegetation returns. This lag reflects the time required to rebuild underground structure. Plants may grow back quickly, but the soil they depend on rebuilds more slowly. Fungi play a central role in closing that gap. When fungal networks are intact, recovery accelerates. When they are absent or disrupted, recovery stalls. This has been observed in studies of reforestation, grassland restoration, and post-agricultural land recovery across multiple continents. There is a gentle irony here. Soil is often treated as something that can be endlessly manipulated without consequence. Tilled, compacted, exposed. But soil remembers. It records disturbance in its structure and chemistry, and it responds accordingly. Healthy soils are not simply present, they are built. They are built by life interacting with minerals over time, guided by physical constraints and stabilized by biological activity. Fungi are among the most important builders in this process, precisely because they operate quietly, continuously, and across scales. Once soil is understood as a self-organizing system rather than a passive medium, the relationship between plants and ground shifts. Plants do not merely occupy soil, they participate in its creation. Through their fungal partners, they help shape the very foundation that supports them. Above-ground stability rests on underground organization. Forests stand tall not only because trees grow upward, but because soil beneath them holds together. Grasslands persist not only because grasses regrow, but because fungal networks bind the ground against erosion. Even agricultural productivity depends, whether acknowledged or not, on soil structures formed long before planting begins. Seen this way, soil is not background scenery. It is a living archive, a slow-moving record of interactions that have unfolded beneath the surface for centuries. Every step across land presses against this archive, every plant draws from it, and every fungal filament adds a line to a history written not in words, but in structure. The ground beneath our feet is not still, it is organized, it is responsive, and it is alive with memory. Cooperation is often imagined as something that requires trust, an expectation that one party will behave fairly, a belief that help given now will be returned later, a sense of shared purpose or mutual understanding. This way of thinking makes sense when cooperation involves humans. It makes far less sense when cooperation involves roots, fungi, and soil. And yet, cooperation clearly exists. Plants and fungi exchange resources reliably across vast spans of time. Nutrients move upward, carbon moves downward. The relationship persists generation after generation. It survives droughts, floods, fires, and climate shifts. It remains stable even though neither participant is capable of trust, awareness, or intention. So how does cooperation persist without any of the mental machinery normally associated with it? The answer lies in how stability emerges from constraints rather than choices. In evolutionary biology, cooperation does not need to be kind, it only needs to last. One of the most influential thinkers on this topic was Robert Axelrod, whose work explored how cooperative behaviors can persist even when individuals attempted to defect. Although his early models focused on simplified interactions, the broader insight that emerged was not about strategy or morality, it was about conditions. Cooperation persists when the structure of interactions makes persistence more likely than breakdown. In biological systems, those structures are not rules or agreements, they're physical and ecological constraints. Plants cannot withhold carbon without consequence. Carbon produced through photosynthesis must move somewhere. If it accumulates in leaves without being transported, photosynthesis slows. Growth stalls. Releasing carbon below ground is not a choice. It is a requirement imposed by internal pressures and transport dynamics. Fungi face a similar constraint. They require carbon to fuel metabolism. Without a steady supply, growth halts and networks collapse. Accessing plant carbon through roots is not generosity on the plant's part, nor manipulation on the fungus's part. It is the most direct way for both to satisfy unavoidable needs. Cooperation emerges because the alternative is failure. This kind of cooperation is best described as constraint-based rather than trust-based. Each participant behaves in a way that sustains the system, because departing from that behavior carries immediate costs. The system stabilizes not through goodwill, but through necessity. This reframes cooperation as something deeply physical. Carbon flows along gradients. Nutrients move where they are most available. Water follows pressure differences. When these flows align in ways that support multiple organisms simultaneously, cooperation appears as a byproduct. No promises are made, no expectations are held. The system simply continues because it works. This explains why attempts to moralize plant-fungus cooperation tend to mislead. Words like generosity, sharing, or reciprocity imply motivation. They suggest that organisms are choosing to cooperate because it feels beneficial or fair. In reality, fairness does not enter the picture. Fungi do not supply nutrients because plants deserve them. Plants do not release carbon because fungi have earned it. Exchange persists because organisms that participated in it left more descendants than those that did not. Over time, this filtering produces systems that look cooperative without requiring any internal sense of cooperation. Evolutionary stability emerges when deviations are penalized by the environment. If a fungal strain extracts carbon but fails to deliver nutrients effectively, the plant's growth suffers. Reduced growth means less carbon available overall. The fungal strain does not prosper. Conversely, if a plant reduces carbon allocation too aggressively, fungal networks weaken, nutrient uptake declines, and the plant becomes less competitive. The imbalance corrects itself through reduced performance. This feedback loop is not conscious, it is mechanical. Stability arises because extremes are disfavored. Moderate, mutually sustaining interactions persist. Over many generations, these interactions become the norm. This process does not eliminate competition. Different fungal species still compete for access to roots. Plants still compete for light and space, even within a single fungal network. Resources are distributed unevenly. Cooperation does not replace competition, it constrains it. The key insight is that cooperation can exist without trust, because trust is not the mechanism that sustains it. Instead, the mechanism is constraint. Physical limits, metabolic requirements, and environmental pressures shape what behaviors are viable. When cooperative configurations fit within those limits, they persist. When they do not, they fade away. This perspective brings a calm realism to how life organizes itself. There is no hidden benevolence guiding underground systems. There is no intention to help or be helped. What exists is a set of interactions that have proven stable across time. Stability, not virtue, is what evolution preserves. Seen this way, cooperation becomes less romantic and more impressive. It does not rely on fragile mental states or shared goals. It relies on the robustness of physical processes. It persists even when conditions fluctuate, precisely because it is grounded in necessity. This also explains why these systems can feel surprisingly resilient. Without trust, there is nothing to betray. Without intention, there is nothing to misunderstand. When cooperation is enforced by constraints rather than expectations, it becomes difficult to disrupt without changing the underlying conditions entirely. Alter the soil chemistry and interactions shift. Change water availability and flows reroute. Remove fungal partners and plants respond accordingly. The system adjusts continuously, not because it is negotiating, but because it is responding. There is a subtle comfort in this. Cooperation without trust does not depend on good behavior. It depends on alignment. When the needs of different organisms intersect in ways that can be satisfied simultaneously, cooperation emerges automatically. This is why plant-fungus partnerships have endured for hundreds of millions of years, not because they are delicate agreements that must be honoured, but because they are robust arrangements shaped by physics, chemistry, and selection. Trust can be broken, constraints cannot be ignored, and so, beneath the soil, cooperation persists without awareness, without intention, and without sentiment. It endures not as a moral achievement, but as a practical one. Life continues, quietly aligned simply because this way of existing works better than the alternatives. It is cooperation stripped of romance, and in that simplicity it becomes remarkably strong. A forest is easy to misunderstand when it is seen only as a crowd. Trunks stand beside one another. Crowns overlap. Leaves compete for light. From a distance, the scene looks like an assembly of individuals, each tree growing according to its own needs and limits, occasionally interacting but fundamentally separate. This way of seeing forests is intuitive. It is also incomplete. When ecologists began to study forests over long periods and across large areas, a different picture emerged. Forests behave less like collections of independent organisms and more like integrated physiological systems. Energy moves through them. Materials circulate within them. Disturbance in one region affects responses elsewhere. Growth and decline unfold in patterns that cannot be explained by individual trees alone. This shift in perspective was central to the development of ecosystem ecology, a tradition strongly associated with the ecologist Eugene Odum. Odum argued that ecosystems should be studied as functional units, with attention paid not just to species composition, but to flows of energy and matter through the system as a whole. In this framework, a forest becomes something closer to an organism. Energy enters primarily through photosynthesis, captured by leaves spread across a vast canopy. That energy is converted into chemical form and distributed through trunks, branches, roots, fungi, microbes, and soil. Along the way, energy is used, transformed, and dissipated as heat. Matter cycles repeatedly. Carbon moves from air to plant tissue to soil and back again. Nutrients are absorbed, stored, released, and reused. These flows are not confined to individual trees. They span the entire landscape. A tree does not photosynthesize for itself alone. Its carbon enters shared pools. Some becomes wood that may persist for centuries. Some fuels fungal networks that connect multiple plants. Some feed soil microbes that influence nutrient availability for the entire forest. In this sense, photosynthesis performed by one tree contributes to the functioning of many. This idea becomes clearer when forests are observed over long time scales. Long-term forest plot research has revealed patterns that only emerge when systems are monitored for decades rather than seasons. Permanent plots, such as those established in tropical forests, temperate woodlands, and boreal regions, track individual trees across generations. Growth, mortality, recruitment, and species composition are recorded repeatedly in the same locations. These studies show that forests exhibit collective behaviour. Tree mortality does not occur randomly. It follows patterns linked to drought cycles, nutrient availability, and past disturbances. Growth rates of different species rise and fall together in response to climate variation. When large trees die, the effects ripple outward, altering light availability, soil moisture, and nutrient dynamics across surrounding areas. No single tree controls these responses. They arise from the structure of the system itself. Below ground, fungal networks play a crucial role in binding this system together. Mycorrhizal fungi link root systems across species boundaries, creating pathways for nutrient and water movement that span entire forest patches. These networks do not connect every tree equally, but they provide a substrate through which resources can be redistributed in response to local conditions. When one area of the forest experiences stress such as reduced water availability, fungal-mediated transport can buffer the effects by redistributing moisture and nutrients from less stressed areas. The process is slow and subtle, but over time it contributes to system-level stability. This is physiology at a landscape scale. In an animal, physiology coordinates the movement of energy and materials between organs. In a forest, coordination occurs through soil structure, fungal networks, and shared nutrient pools. Leaves function like energy-capturing surfaces. Roots and fungi function like absorptive and transport tissues. Soil organic matter functions like storage. The analogy is not perfect, but it is useful. Just as no single organ explains the behavior of an animal, no single tree explains the behavior of a forest. The relevant unit of function is larger than the individual. Eugene Odum emphasized this point by focusing on energy flow rather than species alone. He argued that understanding how energy enters, moves through, and exits an ecosystem provides insight into its organization, resilience, and limits. Species matter, but their roles are defined by how they contribute to these flows. In forests, energy flow is shaped by canopy structure, leaf area, and seasonal dynamics. Material cycling is shaped by litter fall, decomposition, and nutrient uptake. These processes interact continuously. For example, when leaves fall, they carry nutrients accumulated during the growing season. Decomposers, including fungi and bacteria, break this litter down, releasing nutrients back into the soil. Mycorrhizal fungi then mobilize those nutrients and transport them to roots, where they support new growth. This loop operates year after year. Because it is repeated, small changes accumulate. A forest with stable litter inputs and intact fungal networks builds deep, carbon-rich soils. A forest subjected to repeated disturbance loses organic matter and structural complexity. The difference becomes visible above ground as changes in growth rates, species composition, and resilience to stress. Long-term studies have shown that forests often recover from disturbance not by returning to a previous arrangement of individual trees, but by re-establishing system-level function. Species may change, structure may shift, but energy flow and nutrient cycling gradually stabilize again. This resilience is a property of the system, not of any particular species. Scaling the plant-fungus partnership to the level of forests reveals why individual interactions matter so much. Each plant-fungus exchange contributes a small amount to a much larger circulation. Carbon released by one root supports fungi that influence nutrient availability for many roots. Nutrients mobilized in one patch of soil affect growth across the canopy. Over time, these exchanges weave individual lives into a shared metabolism. This perspective also clarifies why forests can behave in ways that seem coordinated without coordination. When drought reduces water availability, transpiration rates across the canopy decline. When nutrients become scarce, growth slows broadly. When conditions improve, growth accelerates again. These responses do not require communication between trees. They reflect shared constraints acting on a connected system. There is a quiet elegance to this. The forest does not need to act as a single organism in a literal sense. It does not need a brain, a center, or a plan. Its coherence arises from the alignment of processes across scales. Local interactions shape global outcomes. Global conditions feed back into local behavior. This is what makes forests both stable and dynamic. They change constantly. Trees grow, die, and are replaced. Fungal networks expand and retract. Soil composition shifts. Yet the overall system maintains a recognizable form because its underlying processes remain intact. Seeing forests as extended physiological systems also shifts how their vulnerability is understood. Damage to individual trees is rarely catastrophic. Damage to the processes that link trees together can be. Disruption of soil structure, loss of fungal diversity, and breakdown of nutrient cycles undermine the system's ability to regulate itself. The result may look like tree loss, but the deeper cause lies underground. This perspective encourages a broader view of conservation and management. Protecting forests is not only about preserving trees, it is about preserving flows, carbon flow, water flow, nutrient flow, the biological infrastructure that allows these flows to remain coherent. There is gentle irony in how this challenges everyday experience. When walking through a forest, it is natural to focus on individual trees. They are tall, visible, and distinct. The system they form together is harder to see because it extends beneath the surface and across time. But it is this hidden integration that allows forests to persist for centuries. From an ecological standpoint, a forest is not merely a place where trees happen to grow. It is a living system with its own internal dynamics, shaped by countless interactions unfolding simultaneously above and below ground. Each tree participates. No tree dominates. The system endures. Once this way of seeing settles in, forests begin to feel less like collections and more like continuities. Not crowds, but processes. Not assemblies of individuals, but expressions of a shared physiology spread across roots, soil, fungi, and air. The forest, in this sense, is larger than its trees. It breathes slowly. It remembers disturbance. And it sustains itself not through control, but through connection. When thinking about how resources move through living systems, it is natural to imagine space first. Roots spread outward. Fungal filaments extend through soil. Nutrients travel from one place to another. Networks connect points across a landscape, and flow seems to be a matter of distance and direction. This picture is useful, but it captures only part of what is happening. In many biological systems, the most important movement is not across space, but across time. Resources are not simply transported from one location to another. They are retained, stored, transformed, and released slowly, sometimes over decades. What matters is not how far something travels, but how long it remains within the system. This shift in perspective changes how plant-fungus partnerships are understood. Fungal networks do connect roots across space, but their deeper role is temporal. They slow things down. They stretch short bursts of availability into long periods of use. They turn fleeting inputs into lasting support. Consider nutrients entering a forest ecosystem. Nitrogen arrives through atmospheric deposition, biological fixation, and the slow breakdown of organic matter. Phosphorus enters primarily through the weathering of rock, a process measured not in seasons but in centuries. Once these nutrients become biologically available, they are immediately at risk of being lost. Rain can wash them away. Gravity can pull them deeper into soil layers beyond root reach. Yet in mature ecosystems, this loss is often surprisingly small. One reason is that fungal networks and soil organisms act as temporal buffers. Nutrients absorbed by fungi are not instantly passed on or discarded. They are incorporated into living tissue, stored in biomass, and released gradually as organisms grow, die, and decompose. This process effectively holds nutrients in circulation long after they might otherwise have been lost. From a plant's perspective, this is not a spatial shortcut. It is a temporal one. Instead of chasing nutrients across ever larger volumes of soil, plants rely on systems that keep nutrients nearby across years and decades. The forest does not need nutrients to move far if it can persuade them to stay. Long-duration ecological experiments have shown how powerful this retention can be. In undisturbed forests, nutrient residence times, the average length of time an atom remains within the ecosystem can span many years. Nitrogen taken up by plants may cycle repeatedly through leaves, wood, litter, microbes, and fungi before ever leaving the system. Phosphorus can remain bound within soil organic matter and fungal structures for decades. This slow cycling creates stability. Short-term shortages are softened because past abundance has been stored. Short-term abundance does not cause chaos because excess is absorbed into biomass and structure. The system behaves less like a pipeline and more like a reservoir. Ecological succession provides a clear illustration of this temporal logic. When an ecosystem is disturbed, such as after a fire or storm, early successional species colonize rapidly. These species grow quickly, capture available nutrients, and begin rebuilding organic matter. Over time, as soils develop and fungal networks re-establish, nutrient cycling slows and stabilizes. Later successional species, often slower growing but longer lived, become dominant. This transition is not just about species replacement. It reflects a shift from fast, leaky nutrient flow to slow, conservative cycling. Fungi are central to this transition. Early in succession, fungal networks are sparse, nutrients move quickly and are easily lost. As succession proceeds, mycorrhizal associations strengthen, soil structure improves, organic matter accumulates, nutrients become increasingly locked into biological cycles rather than escaping the system. This process has been documented in long-term studies across many ecosystems, from temperate forests to tropical landscapes. Experiments that track plots over decades consistently show that mature systems are defined not by faster growth, but by slower loss. Patience in this context is not a virtue, it is a strategy. Biological systems that endure are those that avoid waste. They slow the movement of critical resources, not because speed is impossible, but because speed is risky. Rapid flow increases the chance of loss. Slow circulation increases the chance of reuse. Fungal networks embody this strategy. Their growth is measured in millimeters or centimeters per day, not meters. Their metabolic rates are steady rather than explosive. Their structures persist even when conditions fluctuate. In doing so, they create a temporal scaffold that plants rely on. Carbon provides another example. Carbon fixed during a single growing season does not vanish when leaves fall. Some becomes wood that persists for decades or centuries. Some enters soil organic matter, protected by mineral associations and fungal compounds. This carbon can influence soil structure and nutrient availability long after the original plant tissue has decomposed. The result is a memory of productivity. Past growth shapes present conditions. present conditions shape future growth, the system carries its history forward through stored matter and structure. This temporal continuity is easy to miss because it operates quietly. There's no obvious movement to watch, no dramatic transfer to observe. Instead, there is persistence. Nutrients linger, carbon remains bound, water is held longer in structured soil, life continues drawing on what was built before. This perspective also reframes how stress is absorbed. Drought, for example, is not only resisted through spatial redistribution of water, but through temporal buffering. Soil rich in organic matter retains moisture longer. Fungal networks access thin films of water that persist after rainfall ends. Plants experience not an abrupt loss, but a gradual decline. Again, time matters more than space. Even recovery from disturbance follows this logic. When soil structure is damaged or fungal networks are disrupted, plants may return quickly, but system function lags behind. It takes time to rebuild storage, retention, and slow cycling. Productivity may look normal while resilience remains low. Long-duration experiments have repeatedly demonstrated this lag. Ecosystems may appear recovered above ground while still losing nutrients below ground at elevated rates. Only when slow processes re-establish does stability return. All of this points toward a deeper truth. Life does not merely occupy space, it occupies time. The most successful systems are those that extend their influence across generations, retaining resources long enough to smooth out unpredictability. Fungal networks and plant partnerships are not impressive because they span meters of soil. They are impressive because they span decades of continuity. This emphasis on time prepares the ground for an even broader perspective. When processes are viewed over centuries rather than seasons, different patterns emerge. Stability becomes visible. Accumulation matters more than movement. Small repeated actions outweigh dramatic events. Patience built into structure becomes a defining feature of life on land. Beneath the forest floor, nothing is rushed. Resources circulate slowly. Loss is resisted quietly, and continuity is preserved not by speed but by restraint. Once this temporal view settles in, the underground world begins to feel less like a network stretching outward, and more like a system reaching forward through time. What flows most powerfully is not material across distance, but life across generations. Evolution is often imagined as a steady climb toward independence. Organisms become more complex, more capable, more self-sufficient. Each step forward is assumed to loosen old constraints, allowing life to operate with greater freedom than before. From this perspective, reliance on another organism can look like something to be overcome. But evolution does not work that way. It does not move toward freedom. It moves toward persistence. One of the most important ideas for understanding this is evolutionary lock-in, sometimes discussed alongside the concept of path dependence. These ideas describe how early successful solutions can shape everything that follows, not because they are perfect, but because they work well enough to become difficult to replace. Once a biological system commits to a particular path, reversing course is rarely possible. Evolutionary theorists studying constraint and irreversibility have long recognized this pattern. Stephen Jay Gould emphasized how contingent events early in evolutionary history can determine outcomes far into the future. Stuart Kaufman explored how complex systems settle into stable configurations that resist change. Richard Lewontin highlighted how organisms actively shape the environments that then constrain their own evolution. Together, these perspectives converge on a simple insight. Evolution builds forward on what already exists. It does not start over. The partnership between plants and fungi is a clear example of this principle. When plants first colonized land, they did so in close association with fungal partners. This arrangement solved immediate problems, nutrient access improved, water stress decreased. Survival became possible in environments that would otherwise have been hostile. Once that solution proved reliable, evolution had little incentive to abandon it. Over time, plant lineages evolved within the context of mycorrhizal support. Root systems developed in ways that assumed fungal assistance. Metabolic pathways adjusted to shared nutrient acquisition. Growth strategies incorporated the presence of external networks. This is path dependence in action. Later evolutionary changes did not remove fungal reliance because doing so would have required rebuilding entire systems from scratch. Instead, evolution layered new traits on top of existing ones. Leaves diversified. Vascular tissues became more efficient. Reproductive strategies expanded. But the foundational partnership remained intact. Plants did not outgrow fungal dependence because there was no advantage in doing so. To outgrow the partnership would have required plants to independently evolve the ability to extract immobile nutrients from soil minerals with the same efficiency as fungi. It would have required greater surface area, more enzymes, higher energy expenditure, and increased vulnerability to environmental fluctuation. That path was not favoured because the existing one already worked. Evolutionary lock-in does not imply stagnation. It implies commitment. Once a system becomes integrated, its components adapt to one another. Over time, each part becomes specialized for its role within the whole. Removing one part then causes disproportionate disruption, not because the system is weak, but because it has become efficient. This reframes dependence entirely. Dependence, in this sense, is not a limitation. It is accumulated success. It reflects a long history of reliability. The more generations a partnership persists, the more tightly evolution weaves it into the fabric of life. This is why mycorrhizal associations are so deeply embedded in plant biology. Even plants that appear to operate without fungal partners often retain genetic remnants of the relationship. They may suppress or modify it under certain conditions, but the underlying capacity remains. True independence is rare and usually associated with narrow ecological niches or artificial environments. The hidden partnership is not hidden because it is fragile. It is hidden because it is assumed. When something works for hundreds of millions of years, it fades into the background. It becomes infrastructure rather than feature. Its presence is no longer remarkable, and so it goes unnoticed. Evolutionary lock-in explains why this infrastructure remains. Once plants and fungi became co-adapted, each shaped the evolutionary landscape of the other. Plants influenced fungal diversity by selecting for traits that improved nutrient delivery. Fungi influence plant evolution by altering soil conditions and nutrient availability. The environment itself changed as a result, reinforcing the partnership further. This feedback loop deepened the lock-in. Breaking out of such a loop is not impossible, but it is unlikely. Evolution does not seek novelty for its own sake. It preserves configurations that persist. Change occurs within constraints, not outside them. There is a quiet lesson here about how progress actually works in biology. Progress is not about shedding reliance. It is about stabilizing useful relationships. Systems become robust, not by standing alone, but by embedding themselves within networks that buffer change. This is why the idea of plants eventually becoming fully independent of fungi misunderstands the direction of evolution. Independence would not represent advancement. It would represent redundancy. The partnership persists because it reduces risk. By distributing essential functions across species, the system avoids single points of failure. If one pathway is disrupted, others compensate. If conditions change, flexibility remains. Over deep time, this resilience matters more than autonomy. Evolutionary lock-in is sometimes framed as a constraint that limits future possibilities. In this case, it is better understood as a foundation that supports them. The diversity of modern plant life emerged on top of mycorrhizal relationships, not in spite of them. Forests, grasslands, and agricultural systems all rest on this hidden infrastructure. Seen from this perspective, the partnership between plants and fungi is not an evolutionary leftover waiting to be replaced. It is a core feature that continues to shape what plants can become. The dependence is real, the limitation is imagined. What appears to be reliance is actually a record of long success, written into roots, soil, and fungal networks. It is hidden not because it is small, but because it is everywhere. Evolution did not move past this partnership. It built the world on top of it, and once that is recognized, the title of this story becomes clearer. The partnership is hidden not because it is rare, but because it has always been there, quietly locking life into a path that works. Healthy systems are quiet. They function smoothly enough that their inner workings fade from view. Nutrients cycle, water moves, growth continues. Nothing calls attention to itself because nothing is obviously failing. Paradoxically, it is often only when a system breaks down that its true structure becomes visible. In the study of plant-fungus partnerships, moments of disruption have acted as powerful diagnostic tools. By removing or damaging parts of the system, researchers have been able to observe what those parts were doing all along. The goal of this work has not been to induce collapse for its own sake, but to reveal normally hidden processes by watching what happens when they are interrupted. Agriculture has provided many of these unintended experiments. Modern farming practices often simplify ecosystems dramatically. Fields are tilled, soils are disturbed, fungal networks are fragmented, and chemical inputs replace biological cycling. Crops still grow, sometimes impressively so, but the way they grow changes in subtle and important ways. One of the most consistent findings from agricultural soil studies is that repeated disturbance reduces mycorrhizal abundance and diversity. Tillage physically breaks fungal hyphae. Certain fertilizers reduce the plant's reliance on fungal nutrient acquisition, weakening the partnership over time. Pesticides can alter microbial communities in ways that ripple outward. The immediate effect may be increased yields in the short term. The longer-term effects are slower, quieter, and easier to miss. Plants grown in soils with reduced fungal networks often require higher nutrient inputs to maintain the same productivity. They show increased sensitivity to drought. Their root systems behave differently, compensating for the loss of fungal assistance by investing more energy in direct nutrient uptake. This compensation reveals what fungi were providing. Fungal networks were not optional extras. They were reducing energetic costs, stabilising nutrient access, and buffering environmental variation. When those networks disappear, plants must pay those costs themselves. Soil degradation studies reinforce this picture. In soils that have lost organic matter through erosion or intensive use, structure collapses. Aggregates break apart. Water infiltration decreases. Nutrients leach more easily. Fungal biomass declines, and with it, the glue that holds soil together. The consequences are not always dramatic. Crops may still grow. Grass may still cover the ground. But resilience erodes. The system becomes brittle. Drought that would once have been absorbed now causes stress. Heavy rain that would once have soaked in now runs off. Recovery from disturbance takes longer. By examining these failures, soil ecologists have been able to infer how intact systems function. Researchers such as Ratan Lal have documented how soil organic carbon loss undermines both productivity and stability, not because carbon is valuable on its own, but because it supports biological structure. Elaine Ingham's work on soil food webs has emphasized how disrupting microbial and fungal communities alters nutrient cycling pathways in ways that plants alone cannot compensate for. In forest ecosystems, breakdown has provided similar insights. When forests are clear-cut or heavily compacted by machinery, fungal networks are damaged extensively. Replanting trees can restore canopy cover relatively quickly, but underground recovery lags behind. Studies have shown that replanted forests may take decades to re-establish mycorrhizal diversity and soil structure comparable to undisturbed systems. During this lag, trees grow differently. Nutrient uptake patterns shift. Mortality rates increase under stress. The forest looks restored from above, but functions differently below. This mismatch between appearance and function is one of the clearest lessons of breakdown. The visible system can recover faster than the hidden one. When the hidden partnership is impaired, the system may continue operating, but with reduced efficiency and increased vulnerability. Experimental disruption has also been used deliberately to study these effects under controlled conditions. In some experiments, soil is sterilized to remove microbial life before plants are grown. The results are strikingly consistent. Plants grown in sterile soil show reduced growth, altered root architecture, and increased susceptibility to pathogens. When mycorrhizal fungi are reintroduced, many of these effects are reversed. These experiments demonstrate that what looks like normal growth often depends on unseen support. Breakdown also reveals how finely balanced the partnership is. Not all disruptions cause collapse. Some cause reorganization. When one fungal species declines, others may increase. When nutrient availability changes, carbon allocation shifts. The system adapts within limits. But when disruption exceeds those limits, recovery becomes difficult. Soil ecologists studying system collapse and recovery have noted that certain thresholds exist. Beyond a certain level of organic matter loss, soil structure may not recover easily. Beyond a certain reduction in fungal diversity, nutrient cycling becomes simplified and less resilient. These thresholds are not fixed numbers. They depend on climate, soil type, and biological context. But they illustrate an important principle. The partnership is robust but not indestructible. Recognizing this does not require alarmism. It requires clarity. Plant fungus systems have persisted through ice ages, mass extinctions, and planetary change. They are not fragile in the everyday sense, but they do rely on continuity. When repeatedly disrupted faster than they can rebuild, their stabilizing functions weaken. Breakdown in this sense acts like a stress test. It shows which processes are essential and which are replaceable. It reveals where redundancy exists and where it does not. It exposes how much work is being done quietly in the background when everything seems fine. There is gentle irony in how this knowledge is acquired. The more successful the partnership, the less visible it becomes. Only when it falters do its contributions stand out. Nutrient shortages appear. Water stress increases. Growth patterns shift. What was once taken for granted becomes noticeable by its absence. This pattern mirrors how infrastructure is perceived more broadly. Electricity grids, water systems, and roads attract little attention when functioning well. Their importance becomes obvious only when they fail. The hidden partnership between plants and fungi operates in much the same way. Its breakdown does not immediately end life. It gradually changes how life behaves. Systems become more costly to maintain. More input is required to achieve the same output. Stability gives way to volatility. By studying these changes carefully, scientists gain insight not only into what has been lost, but into what was quietly present before. Breakdown turns absence into information. It reveals that plant-fungus partnerships are not decorative features of ecosystems, they are structural supports. When they weaken, the whole system adjusts, often in ways that are subtle at first and unmistakable later. Understanding this allows recovery to be framed realistically. Recovery is not about restoring appearances alone. It is about rebuilding processes. It takes time to regrow fungal networks, reaccumulate organic matter, and re-establish slow nutrient cycling. Quick fixes can restore productivity, but long-term stability depends on patient reconstruction of the hidden system. This is not a story of inevitable collapse, it is a story of visibility. Breakdown shows where support comes from, it clarifies what matters, it teaches by contrast. When the partnership falters, its role becomes clear, and in that clarity, the quiet importance of what normally goes unseen is revealed. The hidden partnership does its best work when no one notices it at all. Fungi are usually introduced as organisms. They are classified, named and described alongside plants and animals. They have life cycles, reproductive strategies, and ecological roles. All of this is correct, but it also keeps attention focused too narrowly. To understand what fungi actually do, it is often more useful to stop thinking of them as individual organisms at all. Fungi function as infrastructure. Infrastructure is not defined by what it is, but by what it enables. Roads are not destinations. Power lines are not sources of energy. They are systems that allow other processes to occur reliably and at scale. They are mostly invisible when they work well, and painfully obvious when they fail. Fungi occupy a similar position in terrestrial life. They form vast, persistent networks that support energy flow, material cycling, and structural stability across landscapes. They do not simply interact with ecosystems, they help hold them together. This becomes clearer when fungal activity is viewed over long time spans. One of the most significant contributions fungi make to the planet is their role in long-term carbon storage. Plants fix carbon from the atmosphere through photosynthesis, but that carbon does not remain in plant tissue indefinitely. Leaves fall, wood decays. Without stabilizing processes, much of that carbon would return quickly to the atmosphere. Fungi slow this return. Through their involvement in decomposition and soil formation, fungi help convert plant carbon into soil organic matter. Some of this carbon becomes physically protected within soil aggregates. Some binds to mineral surfaces, some is incorporated into fungal residues that decompose slowly. Earth system science research has shown that soils contain more carbon than the atmosphere, and all living vegetation combined. A large fraction of that carbon is stabilized through processes in which fungi play a central role. This does not happen quickly. It unfolds over decades and centuries, one growing season at a time. From a planetary perspective, this matters. Small shifts in how much carbon remains in soil versus returning to the atmosphere can influence climate over long periods. Fungi do not regulate climate in a direct or deliberate way, but through their cumulative effects on carbon storage, they shape the boundary conditions under which climate operates. Mineral weathering provides another example of fungi functioning as infrastructure. Rock weathering is a fundamental planetary process. It shapes landscapes, releases nutrients, and influences atmospheric composition over geological time. Traditionally, weathering was thought of as a purely physical and chemical phenomenon, driven by water, temperature, and erosion. Biology changes that picture. Fungi actively participate in mineral weathering by secreting organic acids that dissolve rock surfaces. This process releases elements such as phosphorus, potassium, and calcium into forms usable by living organisms. Over time, repeated biological weathering contributes to soil formation and nutrient availability across entire regions. This is not a dramatic process. It is quiet, persistent, and cumulative. A single fungal filament dissolves an almost immeasurably small amount of mineral. Millions of filaments doing this continuously over thousands of years transform landscapes. Soil emerges where bare rock once dominated. Ecosystems follow. In this sense, fungi help convert geology into biology. Soil creation itself is a planetary scale process, and fungi are deeply embedded within it. Soil does not appear simply because rock breaks apart. It forms through the interaction of minerals, organic matter, water, and living organisms. Fungi bind particles together, stabilize aggregates, and create the porous structures that allow air and water to circulate. These structures persist long after individual organisms die. They become part of the physical environment, shaping what future life can do. This is infrastructure again. Once built, it supports activity without needing to be rebuilt from scratch each time. It channels flows, it reduces loss, it enables complexity. Earth system science encourages this kind of thinking. Rather than focusing only on individual species or local interactions, it examines how biological, chemical, and physical processes interact to produce stable planetary conditions. In this framework, fungi are not background players. They are active components of Earth's regulatory systems. They influence the carbon cycle. They influence the nutrient cycles. They influence soil formation and water retention. Each influence is modest on its own. Together they shape the habitability of land. What makes this remarkable is the scale mismatch involved. Fungal processes occur at the scale of micrometers. Hyphae are thinner than a human hair. Their chemical reactions affect mineral surfaces measured in fractions of a millimeter. And yet, through sheer persistence and number, these microscopic actions scale upward to influence ecosystems, landscapes, and climate. This scaling does not require amplification in the usual sense. There is no moment where fungi suddenly become powerful. Instead, power emerges through accumulation. Millions of years of steady activity leave marks that cannot be ignored. This perspective also helps explain why fungi are so difficult to eliminate from terrestrial systems. Attempts to simplify ecosystems by removing biological complexity often fail to account for fungal infrastructure. Even when visible fungal fruiting bodies disappear, networks persist underground. Even when networks are damaged, recovery begins as soon as conditions allow. Infrastructure resists removal because so much depends on it. There is a gentle humility in recognizing this. The planet does not rely on dramatic interventions to maintain stability. It relies on countless small processes operating continuously beneath notice. Fungi are among the most important of these processes, not because they act quickly, but because they never really stop. It is tempting to frame fungi as heroic regulators or hidden controllers of the Earth system. That temptation should be resisted. Fungi do not manage the planet, they do not optimize outcomes, they do not correct imbalances with foresight, they simply persist in ways that reshape their surroundings. The regulation that emerges is an outcome, not an intention. This restraint matters. Understanding fungi as infrastructure avoids exaggeration while preserving significance. Infrastructure does not think, it does not decide, it enables. The underground networks that bind soil together, store carbon and mobilize minerals enable plants to grow. Plants enable animals to exist. Animals influence nutrient redistribution. The cascade continues. At each step, fungi remain embedded in the background, quietly supporting the whole. Seen this way, the hidden partnership between plants and fungi extends far beyond individual roots or forests. It reaches into planetary processes that unfold slowly and steadily over time. The soil beneath a forest is not just a local feature, it is part of a global system that regulates carbon, water, and nutrients. Fungi help maintain that system, not through control but through continuity. Scale does not need drama to be impressive. A network measured in micrometers can shape a planet when it's given enough time. Fungi remind us of this. They show how life builds stability not through dominance but through persistence, not through visibility but through structure, not through speed but through patience. As infrastructure, fungi do what the most effective systems always do. They work quietly, they support everything else, and they rarely ask to be noticed. When carbon is discussed, attention usually turns upward. The atmosphere, rising concentrations, gases moving quickly around the planet. This focus makes sense. Atmospheric carbon is easy to measure. It changes on human timescales. It responds visibly to industrial activity and land use. But it is only the most mobile and immediate part of a much larger story. Most of Earth's carbon does not reside in the air. It resides underground. Soils contain more carbon than the atmosphere and all living vegetation combined. This carbon is not static, but it is slow. It moves through biological systems at rates measured in years, decades, and centuries rather than seasons. To understand Earth's long-term carbon balance, it is not enough to track emissions and uptake in the air. It is necessary to follow carbon into the ground and watch what happens there. This is where time becomes the dominant factor. Carbon enters terrestrial systems primarily through photosynthesis. Plants draw carbon dioxide from the atmosphere and convert it into organic compounds. From there, carbon takes many paths. Some is respired back into the air within days. Some becomes leaves or roots that persist for months. Some enters wood that may last for decades. But a substantial fraction moves underground. Through root growth, root death, and root exudation, plants transfer carbon into soil. Fungi intercept much of this transfer. They incorporate plant carbon into their own tissues, transport it through soil, and help convert it into forms that can remain underground for long periods. This process does not eliminate carbon, it slows it. Climate science increasingly recognizes the importance of soil carbon residence time, a concept that describes how long carbon atoms remain stored in soil before returning to the atmosphere. Residence times vary widely. Some carbon cycles back quickly through microbial respiration. Other fractions remain protected within soil aggregates or bound to mineral surfaces for decades or longer. The difference between short and long residence time matters enormously. Carbon that remains underground for a century influences climate very differently from carbon that returns to the air within a year. Over long periods, these differences accumulate. Small changes in soil carbon retention can alter atmospheric concentrations more profoundly than short-term fluxes alone. Earth system models have begun to incorporate this understanding. Rather than treating soil as a passive reservoir, modern climate science increasingly treats it as an active, biologically mediated system. Research from organizations such as the Intergovernmental Panel on Climate Change has highlighted that soil carbon dynamics represent one of the largest uncertainties and one of the largest opportunities in long-term climate regulation. Plants and fungi sit at the center of these dynamics. Fungi influence which carbon compounds persist and which are rapidly decomposed. Through their role in aggregation and mineral association, They help protect carbon from microbial breakdown. Through their own biomass and residues, they contribute chemically complex materials that decompose slowly. This is not a fast solution to climate change. It is not designed to be. Underground carbon processes operate on long timescales because they are constrained by biology and chemistry. They are shaped by growth rates, decomposition rates, and the physical structure of soil. They resist rapid change, both positive and negative. This resistance is precisely what makes them important. Short-term carbon fluctuations in the atmosphere reflect immediate imbalances. Long-term carbon balance depends on slow systems that absorb, retain, and release carbon gradually. Underground processes dominate this long view. Plants alone cannot create this stability. Without fungi, much of the carbon transferred below ground would be rapidly respired or lost. Without soil structure, carbon would wash away or oxidize quickly. It is the partnership between plants, fungi, and minerals that creates durable storage. This partnership integrates carbon into Earth's slow systems. Slow systems are those that change gradually and exert stabilizing influence over time. Ocean circulation, rock weathering, and soil formation are classic examples. They do not respond quickly to disturbance, but they shape long-term outcomes. The underground carbon cycle belongs to this category. Fungal-mediated soil carbon storage does not spike or crash easily. It accumulates quietly. It erodes slowly. It carries the imprint of past ecosystems forward into the present. A forest that grew centuries ago may still influence today's carbon balance through soil organic matter laid down long before living trees were born. A grassland restored decades ago may still be rebuilding underground carbon pools established under previous vegetation. This temporal depth is easy to overlook because it is invisible. Carbon stored in soil does not announce itself. It does not glow or move. It simply remains, altering soil structure, water retention, and nutrient availability in subtle ways. These changes then influence plant growth, which influences future carbon inputs. Time folds back on itself. This understanding also reframes how carbon loss should be interpreted. When soils are disturbed, carbon is often released slowly rather than all at once. Oxidation increases, aggregates break down, residence times shorten. The atmosphere responds gradually, not with a single dramatic pulse, but with a persistent upward drift. Again, this is not a sudden event. It is a quiet shift in balance. Climate science that centers biological storage recognizes this slow feedback. Researchers studying soil carbon, including those working on long-term ecological experiments, have shown that rebuilding soil carbon takes far longer than losing it. Accumulation may require decades of consistent inputs and intact fungal networks. Loss can occur in a fraction of that time if structure is disrupted. This asymmetry is important. It means that underground processes act as long-term stabilizers, but only when continuity is maintained. Once broken, recovery is slow because the system is designed for persistence, not speed. Plants and fungi evolved within this framework. Their partnership reflects a world in which long-term balance matters more than short-term gain. Carbon is not treated as a disposable resource to be used and released immediately. It is integrated into structures that support future growth. From this perspective, carbon cycling is not a loop that closes quickly. It is a spiral that moves slowly forward through time, carrying material from one generation to the next. Atmospheric measurements capture only the most visible edge of this spiral. The deeper motion occurs underground. There is gentle irony in how this challenges modern intuition. Human systems prioritize speed. rapid extraction, rapid release, rapid response. Biological systems that have persisted for hundreds of millions of years prioritize something else entirely. They prioritize staying power. Fungal networks and plant roots do not hurry carbon back into the air. They incorporate it into soil, where it can support life long after the original organism is gone. This does not prevent change, it moderates it. In Earth's slow systems, moderation is power. Understanding this helps place current carbon concerns in a broader context without minimizing them. Atmospheric changes matter. Emissions matter. But so do the quiet processes that determine how much carbon remains part of the living world and how long it stays there. The underground world is where that decision is made. Carbon enters through leaves, but its future is decided beneath the surface. There, plants and fungi work together, not with urgency, but with patience shaped by deep time. They integrate carbon into soil, into structure, into memory, and in doing so, they remind us that Earth's balance is maintained not by fast corrections, but by slow, steady processes unfolding far below our feet, one growing season at a time. Biology often begins by drawing boundaries. An organism is defined by its skin, its bark, its membrane. Inside lies the self. Outside lies the environment. This distinction is practical. It allows measurements to be made, variables to be controlled, and processes to be described without becoming unmanageably complex. But practical distinctions are not always accurate ones. As biological understanding deepens, the idea of an organism as an independent unit becomes harder to sustain. What looks like a clear boundary at one scale dissolves into continuity at another. Cells rely on organelles that were once separate organisms. Tissues depend on signals and substances they do not produce themselves. Bodies exist only within environments that constantly supply energy, matter, and structure. Independence, it turns out, is a useful abstraction rather than a biological fact. Systems biology has been one of the fields most responsible for making this clear. Rather than focusing on individual components in isolation, systems biology studies networks of interaction. Genes are examined not just for what they encode, but for how they regulate and are regulated by other genes. Proteins are understood through pathways rather than functions alone. Cells are modelled as dynamic systems sustained by continuous exchange. In this view, function emerges from relationship. No single part explains the behaviour of the whole. Properties arise because components interact in specific ways under specific constraints. Remove those interactions, and the system ceases to behave as expected. This perspective extends naturally beyond cells and bodies into ecology. Plants do not function independently of soil, fungi, microbes, water, and atmosphere. Their physiology is distributed across these elements. What is often labelled as a plant trait is, in practice, a system-level outcome. A plant's ability to grow tall depends on fungal-mediated nutrient access. Its tolerance of drought depends on soil structure shaped by past biological activity. Its capacity to reproduce depends on conditions created by many other organisms, both living and dead. Seen this way, the plant itself becomes a node rather than a unit. Relational ontology, a philosophical approach that emphasizes relations over substances, provides language for this insight. Rather than asking what something is in isolation, relational ontology asks how something exists through its connections. Identity is not treated as a fixed essence, but as a pattern of ongoing relationships. Applied to biology, this means that an organism is defined not just by its internal components, but by the processes that link it to the world. This does not dissolve organisms into vagueness, it clarifies them. A plant is still a plant, a fungus is still a fungus, but their identities are not sealed. They are permeable, shaped continuously by interaction. Boundaries exist, but they are functional rather than absolute. The illusion of independence arises because many of these interactions are stable. When systems work well, dependencies fade into the background. Nutrients arrive reliably, water is available, growth proceeds. Because nothing demands attention, it is easy to imagine that the organism is managing on its own. This illusion is reinforced by how biology is often taught. Textbooks simplify complexity to communicate core ideas. Organisms are diagrammed as closed systems. Inputs and outputs are acknowledged, but the internal machinery takes center stage. This is effective for learning, but it quietly trains the mind to overestimate autonomy. Real biological systems are far more entangled. Even the most self-sufficient appearing organisms depend on external processes that operate on timescales and scales beyond their control. Plants rely on atmospheric composition shaped by oceans and microbes. Fungi rely on carbon flows produced by photosynthesis. Soil relies on life to maintain its structure. Each dependency is layered on others. Independence is not removed by this recognition. It is redefined. In biology, independence does not mean isolation. It means relative stability within a network of support. An organism is independent to the extent that the relationships it depends on are reliable. This reframing allows dependence to be seen without discomfort. Dependence is not a flaw. It is the natural consequence of specialization. Systems become efficient by dividing labor across components. That division requires connection. Over time, those connections become essential. This is not a moral lesson. It is a structural one. Avoiding moral or social metaphor is important here. The interdependence observed in biology does not imply cooperation in a human sense. It does not imply harmony, kindness, or shared purpose. It reflects physical and evolutionary constraints shaping what persists. Plants and fungi do not rely on one another because they value relationship. They rely on one another because doing so has worked consistently across deep time. This consistency is what gives the illusion of independence its strength. When relationships are stable, they disappear from view. Only when they are disrupted do they become visible. Nutrient shortages reveal microbial contributions. Soil collapse reveals fungal structure. Stress exposes dependencies that were always present. Biology, when examined closely, repeatedly returns to this pattern. What appears autonomous is often supported. What appears isolated is often embedded. What appears self-directed is often constrained. Accepting this does not diminish the individuality of living systems. It enriches it. A plant is not less interesting because it relies on fungi. It is more interesting because its physiology extends beyond itself. Its form reflects not just genetic instruction, but ecological history. Its success reflects not just its own traits, but the persistence of a network that has supported it for hundreds of millions of years. Calm acceptance of interdependence allows biology to be seen more clearly. It removes the need to force living systems into categories shaped by human intuition. It allows complexity to exist without being reduced to simple stories of independence or control. The illusion of independence persists because it is convenient. It simplifies explanation. It aligns with everyday experience. But biology does not require convenience. It requires accuracy. Accuracy reveals a world where life persists through connection. Not connection as sentiment. Not connection as intention. Connection as structure. Once this perspective settles in, the hidden partnership between plants and fungi no longer feels like a special case. It feels like an example of a much broader principle. Life does not stand alone. It stands within. And what appears to be independence is often the quiet success of relationships that have become so reliable they no longer draw attention to themselves. In biology, the self is not a boundary, it is a crossing point. Understanding that does not complicate life. It simply describes it as it has always been. By this point, the partnership between plants and fungi no longer feels like a detail. It feels like a pattern. Not something added onto life, but something life does naturally when given enough time. A way of organizing matter and energy that repeats across scales, environments and histories. Once noticed, it becomes difficult to see biology as a collection of isolated achievements. What stands out instead is continuity. Life persists by staying connected. This partnership teaches that survival is not primarily about strength, speed, or independence. It is about fitting into flows that already exist and shaping them gently enough that they continue. Plants did not dominate land by overpowering it. They settled into it, extended themselves through fungi, and allowed the environment to become part of their own functioning. That approach appears again and again in biology. Cells do not perform every task internally. They rely on organelles, gradients, membranes, and shared chemical environments. Multicellular organisms do not survive by maximizing every function within every cell. They specialize, distribute labor, and depend on internal networks to coordinate activity. Ecosystems follow the same logic. No single species performs all roles. Energy capture, transformation, storage, and recycling are spread across many forms of life. Stability emerges not from redundancy everywhere, but from complementary processes that overlap just enough to buffer change. The plant-fungus partnership fits neatly into this pattern. It is not unusual, it is exemplary. What makes it easy to miss is that it does not announce itself. It does not move quickly. It does not demand attention. Its influence unfolds across seasons, decades, and geological time, shaping conditions slowly enough that cause and effect blur together. This is one of the central lessons it offers. Biological success is often quiet. Processes that persist tend to be those that avoid extremes. They moderate flow rather than accelerate it. They retain resources rather than spending them quickly. They favor arrangements that can absorb variation without collapsing. The underground partnership does exactly this. It slows nutrient loss. It stretches carbon residence time. It stabilizes soil structure. It buffers water availability. Each effect is modest on its own. Together, they create environments where life can continue without constant reinvention. This teaches something important about scale. At small scales, life appears busy. Roots grow, hyphae extend, microbes metabolize. At larger scales, the motion smooths out. Forests persist. Soils deepen. Landscapes stabilize. At even larger scales, continents support life for hundreds of millions of years. The same processes operate at all of these levels. They simply become harder to see as scale increases. The partnership between plants and fungi is one of the threads that runs through all of them. It links the chemistry of minerals to the metabolism of leaves. It links seasonal growth to century-long soil formation. It links individual organisms to planetary cycles. It does so without coordination, without intention, and without any central plan. This absence of control is not a weakness, it is a strength. Systems that rely on control require controllers. Controllers can fail. Systems that rely on constraints persist, as long as the constraints remain. Gravity does not need oversight. Diffusion does not require supervision. Biological processes that align with these forces inherit their reliability. The plant-fungus partnership aligns with physics in this way. Carbon flows because concentration gradients exist. Nutrients move because chemical affinities allow them to. Water follows pressure differences. Evolution shapes structures that take advantage of these tendencies rather than fighting them. Life, at its most enduring, cooperates with the rules of the universe rather than attempting to override them. This idea extends beyond plants and fungi. Symbiosis appears throughout biology because it is an efficient way to build complexity without restarting from nothing. Shared metabolism, shared structure, and shared history allow systems to grow more capable without becoming fragile. Each layer of dependence reduces the need for each part to do everything alone. From this perspective, individuality becomes less about isolation and more about participation. An organism is not defined solely by what it contains, but by what it reliably connects to. The boundaries that matter most are not walls, but interfaces, roots, membranes, surfaces where exchange happens. The partnership teaches that life is not arranged as nested boxes, but as overlapping processes. Once this is recognized, the hidden nature of the partnership takes on a different meaning. It is hidden not because it is obscure or rare, but because it is foundational. Foundational processes tend to disappear from awareness precisely because so much depends on them. Few people think about soil while walking through a forest. Fewer still think about fungal networks, yet those networks make the forest possible. Their success lies in how little attention they require. There is a quiet reassurance in this. Life does not require constant oversight to remain coherent. It does not need to be managed at every level. Given enough time and stable constraints, order emerges naturally from flow. Relationships that work persist. Those that do not fade away. This is not optimism. It is observation. Across hundreds of millions of years, the partnership between plants and fungi has remained because it reduces risk. It spreads essential functions across multiple actors. It embeds life within its environment rather than isolating it from it. It creates resilience through distribution rather than strength through concentration. These are general principles. They apply to cells, organisms, ecosystems, and planetary systems alike. They suggest that the most durable forms of life are those that integrate rather than dominate, that persist rather than accelerate, that connect rather than separate. As this perspective settles, the partnership begins to feel less like a subject of study and more like a lens. Through it, biology looks less like a competition for resources and more like a choreography of constraints. Less like a hierarchy of winners and losers, and more like a continuity of processes that support one another without awareness. This does not remove struggle from the picture. Resources remain limited, conditions change, extinction occurs, but it reframes where stability comes from. Stability comes from relationships that absorb variation quietly enough that life can continue. Preparing for closure does not require drawing conclusions in the usual sense. The partnership does not point toward a moral or a lesson to apply elsewhere. It offers something simpler. It offers a way of seeing, a way of noticing that life's most important work often happens slowly, out of sight, and without instruction. That continuity is built from countless small interactions repeating long enough to matter. That dependence, far from being a flaw, is often the signature of success. The plant-fungus partnership does not ask to be admired. It does not demand recognition. It continues whether it is observed or not, and perhaps that is what it teaches most clearly. Life endures not because it is dramatic, but because it is patient. Not because it is independent, but because it is connected. Not because it seeks control, but because it aligns with what already flows. As the focus begins to soften and the underground world recedes back into invisibility, what remains is not a summary, but a feeling of coherence, a sense that the living world holds together through quiet partnerships that do not announce themselves, but never truly disappear. The hidden partnership has always been there, doing what it has always done, and now, having been noticed, it does not change, it simply continues. Most of the landscapes that feel familiar are supported by work that is rarely noticed. Forests appear stable, fields appear productive, gardens appear simple. The visible world gives the impression of order, achieved through growth that can be seen and measured directly. Trunks thicken, leaves spread, crops rise and fall with the seasons. Yet almost none of this stability originates where the eye naturally rests. The reliability of these landscapes depends on processes that remain hidden by default. They occur beneath the surface, inside soil, within microbial communities, and across timescales that stretch far beyond immediate observation. The visible world is the expression. The hidden world is the support. This is not a poetic framing. It is a scientific one. Soil science, ecology, and earth system research consistently show that the behavior of ecosystems is determined less by what grows above ground than by what happens below it. Nutrient availability, water retention, resistance to erosion, and long-term productivity are governed by underground structure and biological activity. Fungal networks, microbial metabolism, soil aggregation, and organic matter accumulation are the quiet processes that make familiar landscapes possible. When these processes function well, they draw little attention. A forest does not advertise the fungal networks holding its soil together. A meadow does not announce the decades of carbon accumulation beneath its roots. The system appears calm because its internal work is continuous. This continuity lends legitimacy to quiet processes. In science, dramatic events often dominate attention. Fires, floods, droughts, and storms are visible and measurable. They demand response. But the absence of drama should not be mistaken for absence of importance. In many cases, the most consequential biological work happens slowly and persistently, precisely because it must. Soil formation, for example, is not a seasonal phenomenon. It is the outcome of thousands of growing seasons layered on top of one another. Mineral weathering, organic matter incorporation, and structural stabilization occur incrementally. Each year adds a small contribution. Over centuries, those contributions define whether land can support life reliably. Fungal activity sits at the center of this process. By binding particles, storing carbon, and mediating nutrient flow, fungi create the conditions under which plants can grow predictably. Predictability is the foundation of familiar landscapes. Without it, growth becomes erratic. Productivity fluctuates wildly. Recovery from disturbance slows. The legitimacy of these quiet processes is revealed most clearly when they are disrupted. Land that has lost soil structure erodes easily. Fields without intact fungal networks require constant input to maintain yields. Forests growing on degraded soil struggle to recover from stress. The visible changes may be subtle at first, but over time they accumulate into unmistakable instability. These outcomes are not accidents, they are signals. They indicate that the hidden work supporting the system has been weakened. The system still functions, but less efficiently and with less margin for variation. What once absorbed change now amplifies it. Understanding this helps recalibrate perception. A landscape is not simply a collection of plants arranged in space. It is the surface expression of processes unfolding below. What is seen depends on what is maintained. What is maintained depends on work that rarely demands attention, because it has been ongoing for so long. This perspective does not require philosophical expansion. It remains grounded in observation. When researchers study long-term ecological plots, they repeatedly find that surface appearance can be misleading. Two forests may look similar while differing dramatically in soil carbon, fungal diversity, and nutrient cycling. Those differences predict how each forest will respond to future stress more reliably than species lists or canopy measurements alone. The hidden system carries the future. This idea extends beyond forests and fields. Grasslands, wetlands, agricultural soils, and even urban green spaces depend on similar underground processes. Wherever plants grow, they rely on biological infrastructure that precedes them and persists after them. The visible landscape is a temporary arrangement. The underlying system is the constant. Recognizing this does not diminish the beauty of what is seen. It deepens it. A stand of trees becomes more than a collection of trunks. It becomes evidence of long-maintained conditions. A patch of fertile soil becomes more than dirt. It becomes a record of years of biological organization. The calm tone of these processes matters. They are not urgent. They do not hurry. They are not optimized for speed. They operate at rates that match the scales they influence. Soil carbon accumulates slowly because stability requires patience. Fungal networks expand gradually because structure must be reinforced repeatedly to persist. This slowness is not inefficiency. It is design shaped by constraint. Systems that last do not race, they endure. Leading toward closure does not require introducing something new. It requires allowing this understanding to settle. The world as it is seen each day is supported by work that does not announce itself. The ground holds because countless interactions continue uninterrupted. Growth appears reliable because conditions have been quietly maintained. Once this is recognized, attention shifts. The eye may still rest on leaves and branches, but awareness extends downward. Familiar places begin to feel layered rather than flat. Present moments feel connected to past processes rather than isolated from them. This is not abstraction, it is accuracy. A world built on hidden work does not need constant intervention to remain coherent. It needs continuity. It needs time. It needs the quiet persistence of systems that do their job well enough to be forgotten. Science makes this visible without needing to dramatize it. The legitimacy of these processes lies in their outcomes. Landscapes endure, life continues, complexity remains possible. Nothing about this is accidental. The familiar world stands because unseen systems have been holding it up for a very long time, and as attention gently turns toward the end of this exploration, what remains is not a sense of mystery unresolved, but of structure recognized. The hidden work continues, whether it is noticed or not. The surface remains calm because beneath it, everything that matters is still quietly doing its job. Even as attention begins to drift away, the partnership remains. Roots continue to grow into soil. Fungal filaments continue to explore microscopic spaces. Carbon continues to move downward, nutrient by nutrient, molecule by molecule. None of this pauses because it has been noticed. None of it accelerates because it has been explained. The processes that bind plants and fungi together do not respond to observation. They respond only to conditions, and those conditions persist. This is one of the quiet truths revealed by studying long-lived biological systems. Understanding does not complete them. Description does not resolve them. Insight does not bring closure. The partnership simply keeps working. In a forest untouched by recent disturbance, mycorrhizal networks continue to form, break, and reform. Some fungal species thrive, others decline. Roots grow, die, and regrow in slightly different configurations each year. Nutrient pathways shift subtly with moisture, temperature, and plant demand. Yet the overall pattern remains. Plants continue to access minerals that would otherwise remain locked in rock. Fungi continue to receive carbon that would otherwise remain trapped in leaves. Soil continues to gain structure slowly, then lose it slowly, always tending toward a balance shaped by local conditions. This persistence is not static, it is dynamic continuity. Biological systems that endure do so by remaining flexible within constraints. The plant-fungus partnership does not require identical outcomes each year. It requires only that enough exchange occurs often enough to sustain both sides. Variation is allowed, collapse is avoided. From a scientific perspective, this invites humility. Despite decades of research, much of this underground activity remains difficult to observe directly. Measurements capture fragments. Experiments isolate variables. Models approximate flows. Each method reveals something, but none captures the full system in motion. This is not a failure of science. It is a reminder of scale. The partnership operates across centimeters and continents, across days and millennia. It involves chemistry, physics, and biology acting together. No single perspective can fully contain it. What science can do, and has done well, is identify patterns that persist. Mycorrhizal associations appear wherever plants grow naturally. Soil structure improves where fungal networks remain intact. Carbon persists longer underground when biological processes are allowed to operate continuously. Disturbance alters outcomes not by removing life entirely, but by interrupting its slow coordination. These patterns repeat across ecosystems and climates. They do not depend on particular species or exceptional conditions. They reflect general principles of how life organizes itself on land. The partnership persists because it fits those principles. It aligns with energy flow rather than resisting it. It spreads risk rather than concentrating it. It slows loss rather than accelerating gain. There is no final state toward which it moves, no end point to reach, no resolution to arrive at. The partnership is not progressing toward something else. It is not waiting to be replaced by a more advanced solution. It is already sufficient. This challenges a common narrative in science and culture alike. The idea that understanding leads to mastery, and mastery leads to change. In many biological systems, understanding leads instead to recognition. Recognition that some processes endure not because they are fragile or provisional, but because they are deeply compatible with the world they inhabit. Plants did not adopt fungal partners as a temporary measure. They did not retain them out of inertia. The partnership remains because it continues to work under conditions that have changed repeatedly over geological time. Ice ages came and went, atmospheric composition shifted, continents rearranged. Through all of this, the basic structure of the partnership remained intact. That persistence deserves attention precisely because it lacks drama. There is no sudden moment where fungi become important, no threshold beyond which plants suddenly depend on them. The dependence was always there, quietly shaping outcomes. This quietness makes it easy to overlook the partnership when thinking about life in general. Yet it is a useful corrective to how progress is often imagined. Progress in biology is not always about replacement. It is often about refinement. Systems that work are adjusted, extended, and embedded more deeply rather than discarded. Evolution conserves successful arrangements by building around them. The partnership between plants and fungi exemplifies this. It is not a relic of an earlier stage of life on land. It is a foundation that remains active today. Crops, forests, grasslands, and wild ecosystems all rely on it, whether acknowledged or not. Even in environments heavily shaped by human activity, traces of the partnership persist. Fungal spores remain in soil. Networks attempt to re-establish when disturbance subsides. The system seeks continuity whenever conditions allow. This does not mean it is invulnerable. It means it is persistent. Persistence in biology is not the same as permanence. Systems can be degraded. Relationships can be weakened. Recovery can take time. But the underlying logic remains available, ready to resume when circumstances permit. Ending on persistence rather than resolution reflects the nature of the subject itself. There is no final conclusion to draw from a process that has no endpoint. There is only the recognition that life continues to organize itself in ways that reduce waste, distribute function, and embed success into structure. The hidden partnership continues beneath forests, fields, and cities alike. It does so without regard for whether it is named, measured, or discussed. It does not require attention. It does not ask for validation. It does not pause for explanation. It simply keeps doing what it has always done. Roots meet soil, fungi meet roots, carbon meets time, and as long as these meetings continue, life on land retains one of its oldest and most reliable supports. The partnership remains, not as a solved problem or a completed story, but as an ongoing process unfolding quietly beneath the surface. Still working, much of life's work happens without announcement. It does not draw attention to itself. It does not resolve into neat endings. It simply continues, moment by moment, season by season, generation by generation. The partnership beneath plants is one example of this, but it is not unique. It belongs to a wider pattern in biology, where stability is maintained not by visible effort, but by processes that rarely demand notice. The ground holds, nutrients circulate, carbon lingers. Life remains possible because countless small interactions keep aligning quietly enough to endure. What has been explored here is not a hidden intelligence or a secret system waiting to be revealed. It is something more restrained than that. A form of cooperation that does not depend on awareness, a form of organization that does not require control, a form of continuity that persists precisely because it avoids drama. Plants and fungi do not collaborate in the way minds collaborate. They do not decide to work together. They simply exist within constraints that make cooperation the most stable outcome. Over time, that stability becomes structure. Structure becomes expectation. Expectation becomes background. And background is where the most important work often resides. The soil beneath familiar landscapes carries memory. It holds the residue of past growth, past decay, past seasons repeating long enough to matter. What grows today does so partly because of what grew before, and because of what remains beneath the surface from those earlier lives. The present is supported by continuity, not by reinvention. Science, when practiced patiently, makes this visible without needing to elevate it into spectacle. Measurements reveal slow cycles. Experiments reveal dependencies that were always there. Long-term observation shows that what appears calm is often carefully maintained by processes operating just below awareness. There is something quietly grounding in recognizing this. Life does not rely on constant correction to persist. It relies on arrangements that have proven reliable enough to be left alone. When those arrangements are intact, they fade from attention. When they are disrupted, their importance becomes obvious by contrast. Curiosity naturally follows from this recognition. Not curiosity aimed at solving or fixing, but curiosity that notices. That pays attention to what supports the visible world without demanding credit. That allows understanding to deepen without insisting on conclusion. The partnership between plants and fungi does not point toward a final insight. It points toward an ongoing reality, one that continues whether or not it is observed, named, or fully understood. This is not incompleteness. It is continuity. Life on land has been sustained for hundreds of millions of years by relationships like this, unfolding slowly, adjusting quietly, and persisting through change. There is no moment when the work is finished. There is only the ongoing balance between movement and restraint, between growth and holding, between loss and retention. As attention settles, what remains is not a solution, but a steadier way of seeing. The surface world feels less separate from what lies beneath it. Familiar places feel layered rather than isolated. Stability feels earned rather than assumed. Life continues, supported by what does not insist on being seen. And beneath it all, the quiet work carries on, exactly as it always has.